In this section of our video production, we're going to concentrate on the first section of the anatomy notes that will deal with the issues of cytology and histology. And in our first chapter, we're going to be looking at the organelles inside the cell themselves, and we're going to be utilizing basically how these organelles are viewed in the electron microscope in terms of whether they are what are called membrane-bound or non-membrane-bound organelles. We're going to begin first by looking at the first and largest of the membrane-bound organelles, and this is certainly the nucleus, which is certainly typically found in every eukaryotic cell except for a red blood cell. And as we know well from biochemistry, the nucleus is the site of DNA replication, transcription into its precursor RNA molecules. And basically the major elements that we can recognize in the nucleus are different forms of chromatin, euchromatin and heterochromatin, and a fairly typical nuclear inclusion that actually isn't bound by a membrane. This is the nucleolus. The nucleolus is going to be the site of ribosomal RNA synthesis. If we look at the next slide, we can get an overall view of the cytoskeletal or cytoplasmic organelles inside the cell. And again, we've started with the nucleus, and here you can see quite clearly how the nucleus is bound by a double membrane. The unlabeled nuclear inclusion you see on this diagram is the nucleolus. And certainly, in addition to the nucleus, we're going to be dealing with other cytoplasmic organelles outside the nucleus, which consist of the elements of the endoplasmic reticulum, the Golgi apparatus, lysosomes, peroxisomes, mitochondria, and so on. So now, let's look at the first element in the cytoplasm, and these are going to be ribosomes. Ribosomes are largely consist of ribosomal RNA and proteins. They have two component subunits, one small and one large. And basically, these are assembled in the nucleus and transported to the cytoplasm through the nuclear pores. And as we've already alluded to, these subunits are synthesized in the nucleolus, our non-membrane bound nuclear inclusion. Once in the cytoplasm, the ribosomes typically form polysomes, which basically are going to be found in two different forms. And the forms that these polysomes exist in are free polysomes, and these are going to be the site of synthesis of three different kinds of proteins: those destined for the nucleus, those to be incorporated into peroxisomes, or those to be incorporated into mitochondria. Arguably, the more important form of ribosomes are membrane-associated polysomes, and these are going to be the sites of synthesis of three different kinds of proteins: secretory proteins, membrane proteins for the membrane of the cell itself, and most importantly, the lysosomal enzyme proteins. So now, the bound polysomes are actually bound to the rough endoplasmic reticulum, and basically giving it a very characteristic form in the electron microscope, in that rough endoplasmic reticulum typically consists of a parallel array of stacks and sacks of cisternae that are studded with the bound polysomes, typically on the cytoplasmic side. So in the figure to the right at the tip of the arrow, you can see the electron-dense dots that give us the characteristic appearance of the rough endoplasmic reticulum. And as we've alluded to earlier, this is the site of synthesis of the membrane proteins, the secretory proteins, and the lysosomal hydrolase enzyme proteins. And basically, this is the same picture that's illustrated here that, again, makes that point and makes one additional commentary, and that is all of the proteins that are synthesized in the rough endoplasmic reticulum have to be sent to the Golgi apparatus, because as we'll see, the Golgi apparatus is going to be involved in modifying, glycosylating them, and packaging them so that they get sent to the appropriate destination. Smooth endoplasmic reticulum, another membrane-bound cytoplasmic organelle, has the same form of rough endoplasmic reticulum as we'll see, but very different functions. The major thing that we associate at smooth endoplasmic reticulum is steroid synthesis. So certainly it's going to be found in cells in the adrenal cortex, Leydig cells in the testis, and so on. But in addition, smooth endoplasmic reticulum is particularly prominent in liver hepatocytes because it's involved in hydroxylation reactions that involve cytochrome P450, that are basically detoxifying drugs, alcohol, and so on and so forth. Smooth endoplasmic reticulum is also involved in conjugation reactions, where basically there's been a transfer of glucouronic acid from its glucouronic acid UDP to form a toxic water-insoluble molecule. Third, smooth endoplasmic reticulum is also involved in glycogen metabolism, basically freeing or forming the free glucose by cleaving it from glycogen by gluconeogenesis. And then separately, we know that additional reactions that occur in the smooth endoplasmic reticulum involve lipolysis with the release of fatty acids from triglycerides. And finally, we know that smooth endoplasmic reticulum is a specialized component of striated muscle cells that's involved in the storage and release of calcium ions. Finally, if we look at an electron micrograph that's on the next page, we can see the distinct difference in this view of rough versus smooth endoplasmic reticulum. You can see at the top of the slide the parallel cisternal arrays of rough endoplasmic reticulum dotted by electron-dense bound polysomes, whereas immediately adjacent to it, these are most likely liver hepatocytes, are elements of the tubular arrays of smooth endoplasmic reticulum, again looking like the same array of parallel cisternae but lacking the bound polysomes in the form of electron-dense dots. Now then, let's turn our attention to a next membrane-bound organelle, and this is the Golgi apparatus. Golgi apparatus looks for all the world like smooth endoplasmic reticulum in that it is typically found in the form of stacks of parallel arrays of cisternae. The difference is the Golgi apparatus typically has a slight curvature to its membrane-bound cisternae, which you could see in the electron micrograph on the right side of the page. And the other thing that distinguishes Golgi apparatus from smooth endoplasmic reticulum are the presence of dilatations at the ends of the cisternae. So make sure you can distinguish in the electron microscope the difference between Golgi apparatus and smooth endoplasmic reticulum. So the other important point to be made about Golgi apparatus is that Golgi basically got around, and there are several other elements in cell biology that are named after the same person, in that not only did Golgi name Golgi apparatus, but he also named a number of neuron cell types found in various parts of the central nervous system as Golgi cells, such as those in the cerebellum. And also, he, as we look on the next slide, also named the Golgi tendon organs after himself, not to be considered or confused in any way with the Golgi apparatus. So on this particular slide is a diagram illustrating the components and functional areas of the Golgi apparatus. And you can see that the parts of the Golgi are named as the cis portion of the Golgi, the trans portion of the Golgi, and what we call the medial Golgi. Each of these areas have slightly different Golgi-related functions. You could see, for example, that the cis portion of the Golgi apparatus is closely applied to the rough endoplasmic reticulum, and this is typically the point where the newly synthesized proteins generated by the rough ER are entering the Golgi apparatus. In particular, we make a point down at the bottom of the slide. This is where the hydrolase-based enzymes destined for incorporation into lysosomes are entering after being synthesized in the rough ER. The medial part of the Golgi is the glycosylation site, where the Golgi apparatus modifies proteins by glycosylation. And then finally, the trans portion of the Golgi, which is closest to the cell membrane, is the sorting site to ensure that proper proteins get sent and packaged to their proper destination, be they either incorporated into secretory vesicles, as are shown here, to release their secretory products by exocytosis, or that the appropriate proteins, hydrolase-based enzymes, for example, are being sent to lysosomes, or the membrane-based proteins are incorporated into the plasma membrane. So in summary, the functions of the Golgi apparatus include basically a major site of what we call post-translational modification by glycosylation prominently, sorting and packaging of the newly synthesized proteins. The glycoproteins themselves basically are going to be modified in some way to be sent to the proper destination, either as secretory proteins, lysosomal proteins, or the plasma membrane. And two disease processes result from enzyme deficiencies that are missing in the Golgi apparatus. And in both of these cases, the appropriate enzyme-based proteins are sent to the wrong destination or are merely secreted out into the bloodstream. And these two disease states that are Golgi-based are I cell disease and hyperproinsulinemia. In hyperproinsulinemia, there's basically a enzyme defect where there is a missing peptidase enzyme that fails to cleave proinsulin into its insulin and C-peptide subunits in the Golgi apparatus. What in essence happens is there then will be elevated levels of proinsulin in the bloodstream, and patients develop signs and symptoms similar to those with non-insulin dependent diabetes. In I cell disease, under normal circumstances, mannose 6-phosphate residues in glycoproteins are modified by a phosphotransferase enzyme to make sure that hydrolase-based enzymes are sent and incorporated into lysosomes. But when I cell disease results, there is a deficiency in that phosphotransferase enzymes, which means that the lysosomal-based enzymes are sent to the wrong destination. Basically, these enzymes will not be targeted and will not be incorporated into lysosomes, and instead will be merely released into the bloodstream and into other body fluids. And basically, it's the absence of these mannose 6-phosphate residues on those hydrolase enzymes that results in their secretion rather than in their incorporation into lysosomes. And typically, this disease results in a wide variety of skeletal-based abnormalities and a lifespan that's typically less than 10 years. Next, now, let's look at the two different forms of lysosomes. And we've already made the comment about the fact that they are getting lysosomal-based or Golgi and Rough-ER-based hydrolase enzymes of very low pH. We know that lysosomes basically exist in two forms. Primary lysosomes have not yet acquired the materials to be digested or recycled, and they are basically formed most immediately by budding from the trans side of the Golgi apparatus, transphase. Secondary lysosomes are basically involved in some stage of the process of degradation and typically have contents inside their unit membranes that indicate some sort of stage of the degradation process. Lysosomes, as we know, contain mainly hydrolytic enzymes, nucleases, lipases, glycosidases, and so on. All of these are fundamentally acid hydrolases with a pH of approximately 5.0. And we also know that these lysosomal hydrolase enzymes have been synthesized in the rough endoplasmic reticulum. They have been transferred to the Golgi apparatus where they've been modified, sorted, and then ultimately packaged into lysosomes. And we know that lysosomes fundamentally are breaking down large molecules, sphingolipids, glycolipids, and so on and so forth. And there are many of what you'll hear about in pathology, so-called lysosomal storage diseases, based on the fact that there are certain enzyme deficiencies in the lysosomes that result in a buildup of mainly sphingolipids and or their byproducts that are characteristic features of Gaucher's disease, Niemann-Pick's, and Tay-Sachs as well. Finally, electron micrograph, you could see probably the two different forms of lysosomes. The more homogeneous staining that you see kind of in the middle of the micrograph indicates probably the location of primary lysosomes. The arrows are most likely pointing at secondary lysosomes that are in some stage of the degradation process. Now, let's look at peroxisomes. Peroxisomes are actually a specialized form of lysosomes in that in the electron microscope they're very difficult to distinguish, and they have a very specialized class of enzymes that are mainly involved in the metabolism of hydrogen peroxide. Many of the enzymes are basically involved in the formation of hydrogen peroxide, and catalase, the major peroxisomal protein, is involved in the degradation of hydrogen peroxide to its water and oxygen subunits. Peroxisomal enzymes are very different in the sense that, as we alluded to earlier, they are synthesized on free polysomes, and basically after their translation are incorporated directly into peroxisomes without the modification, sorting, and packaging that takes place in the Golgi apparatus. And peroxisomes, in addition to being involved in the metabolism of hydrogen peroxide, are also involved in the oxidation of long-chain fatty acids, the oxidation of residual carbons in the mitochondria, phospholipid exchange, and the elements of bile acid synthesis. And there are a number of genetic-based diseases that are associated with impairment or absence of peroxisomal enzymes. And again, you'll hear about many of these in pathology. The one that comes up with some frequency is Zellweger syndrome. More proper term for it is cerebrohepatorenal syndrome, where basically a whole series of peroxisomal enzymes basically are never incorporated into peroxisomes, or simply degraded as they exist in the cytoplasm. And the way these are described by the exam is patients with Zellweger syndrome have, in the electron microscope, what are known as empty peroxisomes. Neonatal adrenoleukodystrophy, refsums, and hyper are diseases that you'll hear about in pathology. Mitochondria are next, and this, like the nucleus, are cell-based organelles that have a double membrane. Mitochondria are characteristic because their major job is to synthesize ATP and involved in oxidative phosphorylation. And in terms of compartments, the outer membrane of the mitochondrion is smooth, continuous, and highly permeable. But the key element of the mitochondria are the inner membranes that are highly permeable to small ions. These inner membranes are thrown into distinct folds called cristae, which dramatically increase the total surface area of the mitochondrial interior. And as we know well from biochemistry, it is these cristae, these inner membrane foldings, that contain the enzymes for electron transport and oxidative phosphorylation. And fundamentally, we know that since these are the energy sources for a typical cell, based on the level of metabolic activity in a particular cell, that's going to be in direct proportion to the number of mitochondria that are visible and the number of cristae that they contain. And then in electron micrograph, we use this to visualize the Golgi apparatus. You can see surrounding the Golgi apparatus in the middle of the micrograph a number of mitochondria with their distinct internal cristae infoldings. And you can compare that with the schematic diagram of a typical mitochondrion that's illustrated at the insert at the top left of the micrograph. Next, let's turn our attention to the non-membrane bound organelles of a typical eukaryotic cell. And these elements comprise the cytoskeleton. And like many things in cytology, the cytoskeletal elements come in threes. And basically they are form microtubules, which are the largest and most easily visualized of the cytoskeletal elements. Intermediate filaments are next in size, and finally the smallest of the cytoskeletal elements are known as microfilaments. And as we'll see, microtubules are involved in movement of chromosomes because they form the mitotic spindle in dividing cells, regardless of whether they're undergoing mitosis or meiosis. They're involved in intracellular transport, and they provide the mechanism to convey motility to true cilia and flagella. As we'll see in a minute, there are a number of tissue-specific proteins that form intermediate filaments, and we'll go over those in more detail in a minute. Microfilaments, basically the main protein in microfilaments is actin, and certainly the best known place to find actin is where it interacts with myosin in a sarcomere. But as we'll see, actin microfilaments also form the core of microvilli. So now, being more specific, let's look at microtubules first. The major element of microtubules is basically tubulin that has an alpha and beta tubulin polypeptide subunit. And basically, these are polymerized to form microtubules by organizing centers, and also assisted by two accessory proteins, tau proteins, and what are called MAPs, or microtubule assisted proteins or associated proteins. And fundamentally, here are the details about what microtubules are involved in. They are involved, obviously, as we said earlier, in the mitotic spindle, where they play a direct role in chromosomal movement during either stages of meiosis or mitosis. They are involved in intracellular vesicle and organelle transport, and basically this is an energy-requiring process. And as we know from looking at neurons and axons, basically there are two ATPase motor molecules that are driving the transport process, kinesin and dynein. And then finally, as we'll see, dynein is the ATPase motor molecule that's going to convey motility to the microtubule pairs that are found in cilia and flagella. Intermediate filaments, as we said, are intermediate in size between microtubules and microfilaments, and these consist, as we said, of tissue-specific proteins that when their number increases, they may indicate the presence of a tumor-specific kind of marker indicating a specific tumor that's relative to a particular tissue. Most common intermediate filaments include cytokeratins, which are characteristic of epithelial cells, and as we'll see, these kinds of structural intermediate filaments help maintain the integrity of desmosomes and hemidesmosomes, specialized cell-to-cell junctions that are found in a wide variety of epithelial cells. Desmin is an intermediate filament that's characteristic of all different kinds of muscle, gut smooth muscle, and it's also found associated with the Z bands or lines of a skeletal and cardiac muscle. Vimentin is what we call a mesoderm or a mesenchyme intermediate filament. It's associated with endothelial cells, and it's not too much of a stretch to also say that it's also associated with vascular smooth muscle. So when we talk about smooth muscle intermediate filaments, gut smooth muscle is desmin, but the vascular blood vessel smooth muscle is vimentin. But vimentin, as we've said, is also a mesoderm-related intermediate filament that's found in fibroblasts, chondroblasts, and so on and so forth that are associated with the formation of connective tissue and cartilage, the structural support cells of the body. Last place that intermediate filaments are very characteristics are in neurons. Neurofilaments are a prominent feature of all parts of a neuron. And glial filaments are found in virtually all glial cells, but are particularly prominent in CNS-based astrocytes. Finally, microfilaments are the smallest of the cytoskeletal elements, and as we said, they are largely composed of actin. They're fundamentally two different forms of actin, F-actin and G-actin, that are kind of twisted together in a helical pattern. And fundamentally, actin microfilaments are involved in a couple of processes. One is local movement inside the cell itself. And as we know well, the actin filaments are overlapping and associated with myosin filaments in striated muscle cells. They form the core of microvilli that we'll talk about in a minute. And they're also closely associated with the red blood cell membrane. And at that red blood cell membrane, they utilize spectrin as their actin binding protein. Let's now look at a couple of features of the surfaces of a typical eukaryotic cell. And we're going to look at three different aspects of this. This is kind of illustrated by the schematic diagram on this slide. One is at the arrangement in the electron microscope of how the basal membrane, the basal surface of a typical cell, is related to an underlying basal lamina. That's illustrated at the bottom of the picture. Then we're going to look at how cells interact with one another at cellular junctions, and we're going to see that there are fundamentally three different kinds of junctional specializations that have different functions and a distinct feature in the electron microscope. These will be tight junctions, desmosomes, or gap junctions. We'll look at those. And then finally, we'll finish this chapter by looking at the apical or surface specializations of a cell in the form of either microvilli, as illustrated in this picture, stereocilia, or true cilia. Let's begin by looking at the arrangements between the basement membrane, or the base membrane of the cell, and the underlying acellular matrix. And we need to make the distinction between what is a basement membrane and what is a basal lamina. A basement membrane is the light microscopic term and that underlies virtually all epithelia. And recognize that the basement membrane is a light microscopic term. which in the electron microscope can be seen to consist of both a basal lamina and an underlying reticular lamina. The basal lamina is what we pay attention to fundamentally at the electron microscopic level, and it is composed of a specialized form of filtration collagen known as type 4 collagen. It has binding proteins that help keep the cell membrane in question bound to the underlying reticular lamina in the form of laminin, for example, and proteoglycans, which are charged molecules. One of the best known of them is heparin sulfate, found in the kidney basal lamina, that has a net negative ionic charge. The underlying reticular lamina basically is consisting of delicate reticular fibers. And certainly, even though we didn't label it here, the best known and probably most clinically significant basal lamina is that of the kidney glomerulus, which is a basal lamina that, as we'll see when we look at the kidney glomerulus itself, is a basal lamina that's shared by the podocyte foot processes on one side, there to the top right of this electron micrograph over on the right side of this slide. And this basal lamina is also shared by a fenestrated discontinuous capillary endothelium of the glomerular arterioles. Next, let's look at the specializations that occur in the form of junctional complexes that basically are found either uniting one cell to another or help maintain the integrity of one cell and binding it down to the underlying basal lamina. And fundamentally, we can kind of divide up the junctional specializations between cells into three different forms. At the luminal surface of the epithelium, typically will be found a zonula occludens, which is also known as a tight junction. This is only found near the luminal surface of the epithelium, and basically this junctional specialization acts as a barrier because at punctate points, the two opposed membranes of the adjacent cells are basically in direct contact with another in kind of a series of punctate type contacts that provide a barrier protecting the sides of cells from whatever the contents of the lumen of the epithelium are. Next, as we move down is the second kind of junctional specialization. This is known as a desmosome or a macula adherens or a spot-like junction. Basically, this is a junctional specialization that helps keep cells together. And as we'll see, it's going to be prominently associated with a component of intermediate filaments known as tonofilaments, which are highly characteristic of epithelial cells. And then finally, the last junctional specialization is known as a communicator or a gap junction. It's also known as a nexus as well. By communication, this junctional array consists of connexin channels linked together to transmit small hydrophilic molecules or ions, such as calcium ions, and second messengers such as cyclic AMP. So now, other than the functional differences, it's important to be able to recognize what these junctional specializations look like in the electron microscope. And other than their actual location, one of the best ways to distinguish them is on the presence or absence of a particular intracellular density closely associated with them. For example, we note at the top of the page that we can identify relatively easily a tight junction based on its location right near the luminal surface of an epithelium and the fact that there is no adjacent intracellular density associated with the closely opposed membranes. Desmosomes, on the other hand, are the only kind of junctional specialization that have a prominent intracellular density on the cytoplasmic side of the opposed membranes, formed in large part by the associated intermediate filaments. So desmosomes are the most easily recognizable because of this intracellular density of the three junctional specializations. Gap junctions, unfortunately in the electron microscope, look exactly like a tight junction, such that they are closely opposed membranes that contain or consist of, as we noted, the connexin subunits, but they lack the appropriate intracellular density. So the best way to recognize a gap junction is on its location. It's only found between adjacent cells, but never at the luminal surface. Finally, note that a specialized form of desmosome may be situated in the basal surface of an epithelial cell, known as a hemidesmosomes, its job is to help maintain the integrity by joining the basement membrane, the basal membrane of a cell to the underlying basal lamina. In the next picture is an electron micrograph that will help us distinguish between a gap junction and a desmosome. Note that we're identifying this gap junction because it's nowhere near apparently the luminal surface of the epithelium, but you can see it consists of almost a black line in the electron microscope where the closely opposed membranes are situated adjacent to one another. But note there is an absence of an adjacent intracellular density on either side of the opposed membranes. In contrast, look at the appearance of a desmosome. Its membranes are much further apart. There is a much more prominent extracellular space between the opposed membranes, and you can see a prominent electron density caused by the intermediate filaments on either side of the opposed desmosomal membranes. So basically you can see in the electron microscope that these two elements are fairly easily distinguished. Next, let's complete our chapter on cytology by looking at the apical or luminal surface specializations. And again, there are three of these: microvilli, stereocilia, and true cilia. Microvilli themselves are, as we indicate in the text, are luminal specializations whose job overall it is to increase the surface area, particularly of an area like small intestine columnar epithelial cells, specialized for absorption. Note that, as we alluded to earlier, the core of a microvillus is formed by actin microfilaments. And you can see examples of a microvillus epithelium in the micrograph on the right, probably from a columnar small intestine absorptive epithelial cell. But note also you can see examples of some of the junctional specializations at relatively low magnification. Note the appearance of the tight junction or zonula occludens immediately adjacent to the luminal surface of the epithelium, and note how in the electron microscope it looks like an electron-dense black line, basically lacking significant density on either side of the opposed membranes. Compare that with the desmosome that's illustrated a little more inferiorly, where it joins the adjacent epithelial cells together with membranes that are separated by a more prominent extracellular space and are associated with a prominent intracellular density. Note that a zonula adherens is a form of a joining junction that's found just below the area of a tight junction. The function of a zonula adherens is basically to help keep opposed membranes together immediately adjacent to the tight junction. But the zonula occludens, even though it has desmosomal-like functions, is associated with microfilaments rather than intermediate filaments. And since it's associated with microfilaments, you cannot visualize them as prominently as an electron-dense array adjacent to the opposed membranes, unlike the intermediate filaments that form the more prominent density at the level of a desmosome. Now, in contrast, what are stereocilia? We've identified them as another form of a cell surface specialization, and they are certainly that, but recognize that stereocilia are really just tall microvilli found in two specialized locations in the body. They are found at the apices of cells lining male reproductive duct structures, particularly the cells of the epididymis and the ductus deferens, and they form the hairs of hair cells in the inner ear, where in both cases they are non-motile structures, but in the case certainly of hair cells of the inner ear, they are deflected by fluid displacement, not by their own motility. Finally, the other form of specializations that emanate from a cell are known as true cilia, and basically a very good example of a true cilium is a flagella that basically it has the same microstructure as a true cilium, but it certainly is forming the motile tail of a sperm cell. In contrast, a typical cilium, basically as we see on the next micrograph, has an array of microtubular pairs in the form of 9 peripheral pairs that surround a single central pair of microtubules that are kind of linked together by necks and sublinks. And more importantly, these are motile pairs of microtubules because linked, associated with each pair, is a pair of what we call dynein sidearms, dynein being the ATPase motor molecule that conveys motility to that true cilium or to the flagellum of a sperm cell. Functionally, we know that in many cases the cilia basically are beating. They are moving back and forth. Their major job is to propel fluid and particles unidirectionally, and they are certainly an important component of the pseudostratified ciliated epithelium that's characteristic of much of the respiratory system, whose job it is to certainly clear mucus and what mucus contains from the lumina of elements of the respiratory tract. And as we've already indicated, the dynein conveys motility to the flagella, the tails of sperm cells. And finally, a commentary. When there is an absence or a deficiency or a defect in the development of the dynein sidearms, patients develop Cartagener syndrome, where basically they have immotile cilia. And certainly because of their prominent features and presence in the respiratory system, such individuals are prone to chronic sinusitis, bronchiectasis, and so on and so forth. And in males, certainly infertility may be the result because of lack of motility that would normally be conveyed to sperm cells. And during development, it's thought that cilia play a role in the ultimate rotation and position of thoracic and abdominal viscera. So in these patients, newborn patients particularly, they may have backwards or reversed viscera associated with this syndrome. And note again that patients with Cartagener syndrome are not going to have any forms of a hearing problem because, as we noted earlier, the stereocilia, the hairs of hair cells, are not true cilia, even though they certainly are going to move and deflect under fluid displacement, but they're actually tall microvilli instead of being true cilia as a result. So that kind of summarizes our view of the elements of the cell itself in terms of its membrane and non-membrane bound organelles. And remember that it's important that you not only know what those organelles and non-membrane bound organelles do functionally, but it's important for you to be able to recognize what they look like in the electron microscope in terms of their membrane bound components, the density, and electron density associated with them. I'm Dr. White, and now let's continue our tour of histology and cytology with a view beginning with Chapter 2, looking at each of the individual tissue systems of the body. Chapter 2, as we see in this first slide, is going to deal with nervous tissue. And fundamentally, when we talk about nervous tissue, certainly we're illustrating the histology of neurons. And we know from high school biology, for example, that neurons basically have three fundamentally distinct parts. The cell body, which is typical of any eukaryotic cell and found elsewhere in tissues, that it's going to have a typical arrange of cytoplasmic organelles, nucleus, and so on and so forth of a protein-producing cell. The thing that distinguishes neurons from other cell types are its long membrane-bound cytoplasmic processes specialized for conduction of impulses. And certainly a typical neuron is going to have an array of dendrites, and typically a neuron is going to have a single axon. The dendrites are the receptive portion of a neuron, and in a way, the form of a dendritic arbor of a neuron is kind of an indication of what its functional capabilities might be. And as we've indicated, the job of the single axon is to convey action potentials, electrical impulses away from the cell body toward either a target structure or another neuron. Let's look at the cell body first. We've indicated that like a typical neuron, it contains an array of membrane and non-membrane bound organelles typical of any eukaryotic cell. It's going to have a prominent nucleus with a prominent intracellular nucleolus, mitochondria, endoplasmic reticulum, and so on and so forth. Note that in a typical protein-producing neuron, the amounts of rough endoplasmic reticulum are going to be particularly abundant, and when present, this rough endoplasmic reticulum is known as Nissl substance. So recognize that as being a distinct feature of the cytoplasm of a neuron cell body, as well as the proximal parts of a dendrites of a neuron cell body as well. There are also many structural elements to a neuron cell body as well as its axon and dendrites in the form of either microtubules and neurofilaments that form its skeleton. Microtubules are particularly important, as we'll see, in axonal transport, a mechanism whereby precursors and actual neurotransmitters themselves are transported from the cell body where they're synthesized down the length of the axon. And in many cases, neuron cell bodies may be pigmented in the form of containing either lipofuscin or melanin pigments. Melanin pigments are particularly distinct in neurons that are producing catecholamine-based neurotransmitters such as dopamine and norepinephrine. Dendrites themselves, as we know, are the neuronal process that are specialized to be highly branched and transmit information to the cell body. Axons are thin and of uniform diameter, extend from the cell body to whatever the target structure is, be it another neuron or some sort of gland, muscle, or what have you. And as we alluded to earlier, since the axon does not contain rough endoplasmic reticulum, any neurotransmitter or precursor of a neurotransmitter that's synthesized in the cell body must be transported down the length of the axon. And this transport process basically comes in two phases and basically utilizes microtubules. So fast axonal transport is the mechanism most commonly used by neurotransmitters and or their precursors, and it has directionality to it. In fact, it's bidirectional. The direction by which neurotransmitters are transported from the cell body down the length of the axon to the axon terminal is known as anterograde axonal transport. This is an energy-requiring process that's powered by kinesin, an ATPase motor molecule. Retrograde axonal transport, which basically is a mechanism whereby axon terminals can take up and recycle substances, membranes, and the like from the extracellular or synaptic cleft. Retrograde transport will take up those substances and transport substances back along the length of the axon toward the cell body, a mechanism that also utilizes microtubules and is powered by an ATPase motor molecule known as dynein. And certainly at the distal end of the axon are synaptic boutons that participate in specialized junctional complexes known as synapses. And in a typical neuron that utilizes a chemical neurotransmitter, the synaptic boutons will contain different arrays of forms of synaptic vesicles, each with their own quanta of neurotransmitter. In our picture on the next page, you can kind of get the appearance of a typical multipolar neuron. This might be characteristic, for example, of a lower motor neuron whose cell body is somewhere inside the central nervous system, such as in the ventral horn of the spinal cord, and whose axon leaves in a root of that spinal or cranial nerve in this case, and basically goes out and innervates skeletal muscle. The reason we can say that is because in this case the axon emanating from the cell body is myelinated by Schwann cells, meaning that it must be the axon must be in large part found in the peripheral nervous system. You could see just an illustration schematically of a typical neuron of the nucleus, nucleolus. Note that Nissl bodies are illustrated here and consist of prominent arrays of rough endoplasmic reticulum, Golgi for the sorting, packaging, and post-translational glycosylation of neurotransmitters. And you can see that the initial segment of the axon basically is unmyelinated, and immediately distal to that the myelin sheath appears, and as we'll see, there are two different kinds of glial cells that participate in the formation of that myelin. Now, just to review the directionality and the elements involved in the transport process, you can see in this illustration that anterograde axonal transport is mediated by dynein and also is certainly facilitated by microtubules, whereas retrograde axonal transport, basically associated with recycling of neurotransmitter and membranes, is occurring in the opposite direction, moving substances from the axon terminal back to the lysosomal system in the cell body. Retrograde axonal transport, as indicated here, as we've alluded to, is mediated by dynein. Next, let's look at a view of the three different fundamental ways of staining neural tissue to demonstrate it histologically. And they're kind of illustrated on these three panels in the middle of the slide. Two common ways of illustrating neural tissue are shown on either ends of these. One is to use a so-called Nissl stain, which, as we know, will only react with rough endoplasmic reticulum and therefore will only stain basically the cell bodies of neurons and or glial cells, as well as the proximal parts of the dendrites. So in the image on the left, you see basically the appearance of an area, in this case of cerebral cortex, where basically the Nissl stain is capable of demonstrating the difference in a way, the size and particularly the shape of particular populations of neuron cell bodies, and it also shows their degree of layering or lamination. In the middle panel is a way of demonstrating all of the processes as well as the form of a cell body of a neuron, and this is basically a method that stains a very small population or percentage of neurons in a particular section, utilizing a silver method known as the Golgi method. When it works, and nobody fundamentally understands how it works, it demonstrates not only the neuron cell body, but all of its processes, including the origin of the axon. Third, the other common way of staining neural tissue is simply to react it with what's called a myelin stain, which certainly will only stain axons that are myelinated. It's also known as a fiber stain that will simply illustrate just myelinated fibers and no parts of either the cell body or its dendrites. Some examples of this are shown on the next two slides. First one is a section through the spinal cord that's stained with a Nissl stain. And very clearly you can see in the area at B are some very large neurons visible even at this low magnification in what we refer to as the gray matter of the interior of the spinal cord. In particular, these large neurons are lower motor neurons that are characteristic of the ventral horn of the gray matter of the spinal cord. Outside them is an unstained area indicated by letter C. We call it white matter, and as you can see in a particular Nissl stain, it's unstained because basically the area at C completely surrounding the gray matter, it basically contains nothing but bundles of axons. In contrast, using a myelin stain on the next figure, also a section through the spinal cord, we can see that the area at A, where the myelinated axons are found, are very heavily and darkly stained throughout the area and surrounding the spinal cord, whereas the area at B is largely unstained because this was the area of the gray matter that almost exclusively consists of neuron cell bodies, their dendrites, and the only light staining that you see in the area of B are axons, myelinated axons, that are either entering or leaving cell bodies in that area. Next, let's look at a specialized area associated with nervous tissue, and this is the area of axonal specialization at the neuromuscular junction. And this is the specialized junctional array that's found between the distal end of a motor axon of a lower motor neuron and its distinct features where it synapses with a muscle cell. And basically, at this neuromuscular junction, the synaptic cleft is thrown into a series of subneural clefts or folds, and basically this area is where acetylcholine is released from the axon terminal of the motor end plate and basically interacts with nicotinic cholinergic receptors on the postsynaptic membrane. And in this figure we can see that illustrated are some of the details associated with a neuromuscular junction. Specifically, you can see the axon nerve terminal releasing synaptic vesicles containing acetylcholine, and you can see basically where they interact with postsynaptic receptors in the junctional folds, which are nicotinic cholinergic receptors. Also, what's shown on here are a number of the different clinical elements that affect neuromuscular junction or the axons that lead to them. Specifically, they include peripheral myopathies that involve myelin of axons near the neuromuscular junction that are affected in Guillain-Barré syndrome. And also, what's illustrated on here are other problems that affect the neuromuscular junction, such as the antibody binding to cholinergic receptors that's a cause of myasthenia gravis, the area where botulism toxin binds to the presynaptic membrane, the site where curare basically binds to the cholinergic receptors themselves, as well as the antibodies that are raised against presynaptic calcium channels that is a cause of the Lambert-Eaton syndrome. And the details of these deficits are summarized kind of on the next couple of slides. Fundamentally, when we deal with disorders of neuromuscular transmission, there are a variety of disease states and causes of them, including botulism, myasthenia gravis, curare, snake venom, and Lambert-Eaton syndrome, and some of them are summarized clinically in the next couple of slides. Botulism toxin basically prevents the release of acetylcholine by binding to the presynaptic membrane. And basically, these patients have weakness initially in muscles innervated by cranial nerves. Then finally, they're also going to have autonomic signs and symptoms, including dry mouth, abdominal cramping, vomiting, and diarrhea due to basically a problem at the location of junctional specializations associated with smooth muscle. Next, in patients with MG, myasthenia gravis, typically this is caused by antibodies that bind to the cholinergic receptors, therefore blocking the binding ability of acetylcholine. And classically, these patients have weakness initially in skeletal muscles innervated by cranial nerves. Particularly in most of these patients, they will have ocular weakness exhibited basically by a bilateral ptosis. And then fundamentally, most of these patients have an underlying thymoma or thymic hyperplasia. And overall, sustained contractions of the muscles affected in these patients typically leads to increased weakness. And patients typically present more likely with the signs and symptoms as they move along in their day and basically get tired. Curare and alpha-bungarotoxin have the same basic effects. They also bind to the cholinergic receptors and block neuromuscular transmission. In essence, having effects similar to those in patients with myasthenia gravis. Patients with LEMS, basically an underlying neoplasm is typically present in these patients. In this case, however, antibodies are raised against the presynaptic calcium channels that disrupt neuromuscular transmission and the release of acetylcholine. Skeletal muscles, again, in the limbs in these patients are primarily affected, as opposed to skeletal muscles innervated by cranial nerves affected in MG. In contrast, however, in patients with LEMS, sustained contractions of affected muscles leads to an increase in strength rather than to a decrease in muscle strength. Next, let's look at the different forms and glial cells that make myelin and the effect of myelin on its appropriated axons. Certainly, axons in the peripheral and central nervous system may or may not be myelinated, depending on, obviously, their functional capability. And fundamentally, when we see or visualize unmyelinated axons, and when you see them in an electron micrograph, for example, they're very distinct in the sense that unmyelinated axons in peripheral nerves are always encompassed and surrounded by the cytoplasm of Schwann cells. Schwann cells, as we know, are the cells, the glial cells in the peripheral nervous system that make peripheral nervous system myelin. In contrast, when we visualize unmyelinated axons in the central nervous system, they are simply what we consider to be bare axons, and they're not surrounded by the cytoplasm of any corresponding glial cell. And we certainly know from physiology the value of a myelinated axon. The myelinated axons certainly are going to be larger in diameter, but more importantly are faster conducting than unmyelinated axons. The two different forms of myelin-producing cells are Schwann cells in the peripheral nervous system and oligodendrocytes in the central nervous system. Very different feature about how much myelin they make, however. A single Schwann cell is only going to be able to myelinate a single segment of a single axon in a peripheral nerve. So just remember that Schwann means single in this case. Oligodendrocytes, which are the myelin-forming cells of the central nervous system, basically are capable of myelinating many segments of many individual axons adjacent to an individual oligodendrocyte. So a single oligodendrocyte is myelinating many segments of many axons, whereas a Schwann cell is only myelinating a single segment of a single peripheral nerve axon. Next, in both the central and the peripheral nervous system, where there are discontinuities between the segments where there is a myelin sheath, what is created is known as a node of Ranvier. Nodes of Ranvier basically are where the membrane of the naked axon is directly exposed to the extracellular space, and this is a site where saltatory conduction takes place because there are there is a prominent concentration of sodium channels located in the membranes at the area of a node of Ranvier. Again, in a peripheral nerve micrograph, we know this is that of a peripheral nerve because not only do we see the distinct electron-dense appearance of the myelin sheath, but we know this isn't a peripheral nerve because we see in close proximity to two of these myelinated axons the cytoplasm and the prominent nucleus of Schwann cells. And this is a distinct difference from what we would see if we saw a tract of axons inside the central nervous system, where we would be considerably less likely to see any related glial cell associated with them. So now our next comment bears in mind what happens to an axon that's been severed or injured in the peripheral versus the central nervous system. In our examples here, we're indicating that there's been a traumatic site of an injury that, in essence, has severed an axon somewhere in the peripheral or the central nervous system. So when you cut an axon in the peripheral or central nervous system, basically you get two forms of degeneration. You get what is known as anterograde or Wallerian degeneration, where all parts of the axon distal to the cut completely degenerate because they've been cut off from their site of nutrition, obviously the cell body. All axons basically proximal to the cut nearest the cell body undergo what are known as retrograde degenerative changes, basically that are most evident in the cell body itself, which is going to make an attempt to regenerate the cut axon. Now, the point that we need to make is only axons that are myelinated by Schwann cells will actually have the capacity to regenerate. An axon anywhere that's severed or injured in the central nervous system that's myelinated by oligodendrocytes does not have the capacity to regenerate. So the points that's illustrated here is, regardless of where the axon is cut, the axon will generate sprouts that will attempt to regenerate the axon, but only axons that are myelinated by Schwann cells will actually have the capacity to promote the regenerating axon, because the Schwann cells, particularly the endoneurium of the Schwann cell, basically will remain intact and provide a sheath to guide the regeneration of the cut axon, basically guiding it back to its original site where it can reinnervate, for example, in this case, an area of skeletal muscle. So the point is, only axons that are myelinated by Schwann cells have the capacity to regenerate, and in order for the axon in the peripheral nerve to regenerate, the endoneurial sheath, which is in essence a component of the membrane of the Schwann cells, has to be intact to promote and guide the growth of the regenerating axon. And finally, we know that there are two different forms of demyelinating diseases associated with axons whose myelin is formed by either Schwann cells or oligodendrocytes. Oligodendrocytes, when they are affected by a so-called central nervous system demyelinating disorder, the most common of these disorders is known as multiple sclerosis. And basically, multiple sclerosis typically affects myelinated axons in the central nervous system that are found in tracts or fasciculi near the surface of the spinal cord, brainstem, or near the surface of a particular portion of a ventricle. And since this is a central nervous system demyelinating disease, the only nerve that's commonly affected by multiple sclerosis is the optic nerve. Next, let's look at neuroglia, because these cells are what we call the connective tissue or the support cells of the nervous system, because the nervous system in essence lacks connective tissue. There are a wide variety of neuroglial cells. We've already alluded to two types of them. They are oligodendrocytes and Schwann cells respectively that are producing different forms of myelin. But in addition, there are microglia, astrocytes, and ependymal cells that also play a role in different ways as glial cells in the central nervous system. Astrocytes are the most numerous of the glial cells. Typically, they play a role in the overall support of the central nervous system. By their vascular end feet, they contribute to maintenance of the blood-brain barrier, and they also take up excess neurotransmitter and ions in helping to maintain the extracellular microenvironment of nerve cells. Oligodendrocytes, as we've alluded to already, basically their job is to myelinate many individual segments of many different axons exclusively in the central nervous system. They're affected by a central nervous system demyelinating disorder like multiple sclerosis. Microglia are basically the phagocytes of the central nervous system. They're very different in a sense that they're not derived from neuroectoderm, which gives rise to all other glial cells and neurons in the central nervous system. These are cells that are derived from mesoderm that migrate into the central nervous system. They basically are phagocytic cells, part of the mononuclear phagocyte system. These are the cell types that are affected by HIV type 1, and basically HIV does not actually kill nerve cells themselves, but basically it infects the microglia and causes them to produce cytokines that are toxic to neurons. Finally, ependymal cells line the ventricular cavities of the brain and the central canal of the spinal cord, and their job is to maintain the integrity of the ventricular system. And where the ependymal cells are invaginated by choroidal blood vessels, these areas of ependymal cells become modified and form choroid plexus, in particular areas of parts of the ventricles of the CNS that are involved in the production and secretion of cerebrospinal fluid. Finally, Schwann cells are, as we alluded to, the cell types that produce peripheral nervous system myelin. They're affected in peripheral neuropathies like Guillain-Barré syndrome that affects basically myelinated axons in all cranial and spinal nerves, mainly causing motor weakness as opposed to sensory loss. This completes our summary of Chapter 2, the nervous system, which basically reviewed the elements of neural tissue, mainly concentrating on neurons and their processes, obviously describing the elements of the cytoplasmic features of neurons, axonal transport, as well as the different forms of glial cells that myelinate axons, and then looked at the other different forms of glial cells, astrocytes, ependymal cells, and microglia that play a role in central nervous system function. Let's now continue our tour of histology by examining the different elements at the both light and electron microscopic level of muscle tissue. And in our first chart under muscle tissue is a useful comparison chart that illustrates the basic form of the three different kinds of muscle: skeletal, cardiac, and smooth. Note first off, as we'll see, that both skeletal and cardiac muscle are considered to be striated, in that, as we'll see, they have a very distinct banding pattern of overlapping myosin and actin filaments. Note, however, that skeletal muscle fibers are unbranched and separate, whereas cardiac muscle fibers are highly branched and, as we'll see, are going to be interconnected with one another by specialized junctional complexes. Smooth muscle, even though it contains actin and myosin filaments, is basically non-striated and not organized into the sarcomere banding pattern that's characteristic of skeletal and cardiac muscle. Note, in addition, at the light microscopic level, skeletal muscles are multinuclear. The nuclei typically basically found on the outside or the perimeter of skeletal muscle cells. Cardiac muscle cells typically contain a centrally located single nucleus. Smooth muscle cells also a single nucleus. Skeletal muscle cells physiologically are associated with strong, quick, discontinuous voluntary contraction because of their innervation directly by lower or alpha motor neurons. Cardiac and smooth muscle, in contrast, are under control of the reflex elements largely generated on the motor side by elements of the autonomic nervous system. If we look fundamentally at the cross-section and kind of a longitudinal view of skeletal muscle, we can see that its individual myofibrils, as indicated, are not highly branched and are separate. The entire muscle itself is enclosed by a connective tissue investment of the epimysium, whereas the individual muscle cells themselves are all surrounded by an individual connective tissue coat of the endomysium. Now, if we move on to look at the difference in the ultrastructure of skeletal versus cardiac versus smooth muscle, this is also illustrated on this chart. Notice we've already indicated that striated muscle, skeletal and cardiac, can contains or consists of a distinct overlapping banding pattern of actin and myosin filaments, whereas even though smooth muscle contains actin and myosin, it does not form a distinct banding pattern. Note that all three kinds of muscles have elements of an endoplasmic reticulum, a sarcoplasmic reticulum, which is associated with the movement and storage of calcium, but only skeletal and cardiac muscle have a distinct T-tubule system associated with the sarcoplasmic reticulum. Differences being skeletal muscle has a more well-developed series of T-tubules that form a triad with adjacent elements of the sarcoplasmic reticulum. These elements could be diagnostic in the electron microscope for a skeletal muscle sarcomere because the triads of the T-tubule system are found at the AI band junction in the sarcomere. The T-tubule system in cardiac muscle is a little less well developed, only forming what are called dyadic contacts with smooth endoplasmic reticulum that are found near the margins of a sarcomere at the Z-line. Smooth muscle has only limited amounts of sarcoplasmic reticulum and lacks a T-tubule system altogether. Other than the fact that skeletal and cardiac muscle sarcomeres look exactly alike, and the only way thus far we can distinguish them is the presence and location of their T-tubule system, the best way to distinguish skeletal from cardiac muscle is the presence of junctional specializations that are found between individual cardiac muscle cells known as intercalated discs in the electron microscope. These intercalated discs form a complex of desmosome-like contacts that keep the branched cardiac muscle cells together, interposed between gap junctions that promote cell-to-cell communication. In contrast, skeletal muscle sarcomeres lack any semblance of such junctional complexes, making it easy to recognize cardiac muscle cells, as we'll see in the electron microscope. Gap junctions are a prominent feature of the smooth muscle cells, but smooth muscle cells do not have the other components of an intercalated disc, the desmosomal-like contacts. The calcium-binding proteins are different in striated versus non-striated muscle cells. Troponin C acts as the calcium-binding protein in both skeletal and cardiac muscle. Calmodulin is the calcium-binding protein that's present in smooth muscles. And then the intermediate filament of striated muscle, in all cases, is desmin, whereas the intermediate filament that's basically present in smooth muscle in the gut is desmin, but in vascular smooth muscle, it's vimentin. Now, if we actually look at a diagram and then some electron micrographs of the banding pattern associated with striated muscle, we can see that a sarcomere is defined as being the distance between the Z lines. And you can see the Z lines illustrated at both the figure above and the diagram in the middle of the page. Note that the actin microfilaments are the filaments that are actually attaching to the Z line, and that the I band on either side of the Z line exclusively contains parts of the actin microfilaments. In the middle of each sarcomere, not bound or closely associated with the Z line, are the A bands, and they consist exclusively of the thick filaments that basically consist of myosin. Finally, the H band, as you see, is the zone between the ends of the actin microfilaments. And a point to be made that is when the muscle contracts and the sarcomere shortens, it's important to remember which bands are not going to change their length. Certainly because the A band corresponds to the entire length of the myosin filaments, it's not going to change when the sarcomere shortens. But the I band certainly is going to change its length as the sarcomere Z lines come closer together and the overlapping between the myosin and the actin filaments becomes more distinct. And then finally, the H band, which as we alluded to is the point between the ends of the actin filaments, is also going to change length as the sarcomere shortens and the actin microfilaments come closer together. In an electron micrograph, we'll be able to see this even more distinctly. So here's the point. Sarcomeres consist of thick and thin filaments that are overlapping. The thick filaments, as we said, are centrally located, basically in the middle of the sarcomere, and they correspond to the A bands. The I bands basically contain the thin filaments of actin. They are attached to the Z line, and basically the H band corresponds to the area of the middle of the sarcomere between the ends of the actin microfilaments, and basically contains only the central portion of the myosin filaments, respectively. Now let's look at the differences in the banding pattern of sarcomeres as found in both skeletal muscle and cardiac muscle electron micrographs. Certainly when you look at these, it's going to be very important to recognize which bands are which. Note that the most distinct band basically in an electron micrograph of a sarcomere, the most electron-dense band corresponds to the Z line. And you can see them very distinctly illustrated here. The light area of lack of electron density immediately adjacent to that electron-dense Z line is the I band, which, as we know, consists only of the actin microfilaments. The A band, basically filling up much of the central aspect of these sarcomeres, corresponds just to the area of the myosin filaments, but certainly is overlapped as well by the corresponding thin filaments. And then the H zone is basically an area that consists only in the central part of the sarcomere of myosin filaments only. Finally, let's look at a higher magnification view. It illustrates again quite well the location and the different forms of these bands and their configuration in the electron micrograph. This again is skeletal muscle. Note the distinct appearance of the Z line and how it basically bisects a lighter staining zone in the electron micrograph, which, as we've alluded to several times now, is the I band, the area of the thin actin filaments. Note that the more electron-dense component of the sarcomere corresponds to the A band, where the thick myosin filaments are found. Certainly these are overlapped by the thin filaments as well, but the A band, by definition, just corresponds to the entire length of the myosin filaments. And the H zone, as we've alluded to, contains myosin only. It's an area between the distal ends, or the central ends, of the myosin filaments. And then the M line is basically where the myosin filaments are being linked together, respectively. And note also, even though we haven't labeled on here, you can see elements of the T-tubule array and the sarcoplasmic reticulum on either side of these corresponding skeletal muscle sarcomeres, noting that even though it's not immediately obvious here, the elements of the T-tubule system in skeletal muscle would be located at the AI band junction. Now compare this to an electron micrograph in our last slide of a view of cardiac muscle. Note that the banding pattern basically looks, in essence, very similar. You can see the prominent electron density that's characteristic of the Z lines. You can see the letter Z situated close to a Z line. And basically, you can't really see well the distinct area of the I band, but you can see the A band and the overlapping and the central portion of the sarcomere, the M zone, and the H band in the center of the sarcomere. What characterizes this, certainly as cardiac muscle, is not the sarcomere banding pattern. It's the presence of the prominent intercalated disc, which is, as we alluded to, an array of specialized junctional complexes that are linking adjacent cardiac muscle cells together. And you can see that this intercalated disc in the electron microscope has two components to it. It has closely associated membrane arrays situated adjacent to prominent intracellular densities. These are where we have the fascia adherents, specialized desmosomal-like contacts that help keep the highly branched cardiac muscle cells together. At the tip of the black arrow, you can see an electron-dense line that indicates certainly the location of a gap junction, the other important element of an intercalated disc visualized in the electron microscope. So it is this intercalated disc array of specialized junctional complexes that's very characteristic and makes cardiac muscle cells easily identifiable and distinguishable from the sarcomere banding pattern characteristic of skeletal muscle. In summary now, we've basically briefly reviewed the different ultrastructural characteristics and the banding pattern of skeletal muscle, cardiac muscle, and smooth muscle, noting that all three types have elements of actin and myosin filaments, but only skeletal and cardiac muscle have the distinct sarcomere-like banding pattern, and that cardiac muscle can be easily distinguished from skeletal muscle in the electron microscope by the presence of the components of the intercalated disc. Let's now turn our attention to Chapter 4, which deals with the differences in the histology of the lymphatic organs. In our first slide, basically, we compare and contrast the two different forms of lymphatic organs. A primary lymphatic organ basically is found in either the bone marrow or the thymus. And basically, this is where the associated lymph cells or lymphoid organ cells, B cells and T cells, are either made or maturing. Secondary lymphatic organs include lymph nodes, nodules, which obviously are associated with them, and the spleen. And this is basically where the mature T cells, B cells, and or antigen-presenting cells migrate and interact there, basically, and become involved in an associated immune response. First off, let's look at the thymus. And one of the things we're going to see when we deal with lymphatic organs is some structural differences, which we can use to distinguish them. And we can utilize the presence or absence of germinal centers as obviously an indicator of structure. The thymus, being a primary lymphatic organ, is not going to have any germinal centers, but it, like a lymph node, is going to be well structured and basically has distinct cortical and medullary regions. Arguably, the most important area of the thymus is in the area of the cortex, because this is where the immature thymocytes are going to be found, and it is the location of what we call the blood-thymus barrier, where the immature thymocytes are protected from involvement of the immune system. Basically, the thymus itself not only contains a distinct cortical and medullary region, basically its major cell type other than the thymocytes are epithelial reticular cells, Hassel's corpuscles, which are probably degenerative remnants of those epithelial reticular cells that are found in the medulla. We said certainly as a primary lymphatic organ, the thymus lacks germinal centers, and its developing T cells are protected in the cortex by a blood-thymus barrier that consists of capillary endothelium, connective tissue, and underlying basal lamina, and basically the cytoplasm of the adjacent epithelial reticular cells themselves. Basically, the functions of the thymus are, particularly in the area of the cortex, it's a site of T cell maturation where the thymocytes learn to distinguish self from non-self, and only the thymocytes that are tolerant of self-antigens basically are selected and allowed to migrate down to the medulla, basically to pass out of the thymus into secondary lymphatic organs, be the vascular circulation. The self-reactive thymocytes, which basically will be the majority of the thymocytes, die basically through programmed cell death or apoptosis. Lymph node, now being a secondary lymphatic structure, is associated not only with the filtration of lymph containing both afferent and efferent lymphatic blood vessels, but it also has a distinct blood supply that's delivering lymphocytes to it and from it as well. The lymph node, like the thymus, has a distinct cortex and medullary region, and as we'll see, the cortex is further divided into an outer cortex, which is going to be B cell and germinal center rich, and an inner or paracortical region, which as we see is going to be a T cell rich region, and an association medulla. As we've already indicated, the outer cortex contains the nodules and the germinal centers. It's populated obviously by B lymphocytes. The inner cortex or paracortex is going to be the T cell rich area of the corresponding lymph node. In a diagram, we can certainly see this, and using some schematic illustrations, what you see again is the area of the outer cortex containing the B cell rich germinal centers, the inner or paracortex being the T cell rich portion of the lymphocyte, of the lymph node, and then finally we see the adjacent medulla where the efferent lymphatic blood vessels and obviously the modified B cells, plasma cells, and so on and so forth are exiting. Note that the flow of lymph through the node goes from cortex to medulla, but obviously the flow of blood is basically all medullary in origin and exit. So note that the paracortex, being the T cell rich region, will be the part of a lymph node that will be smaller in size in patients that develop the DiGeorge syndrome. Basically, this is a pharyngeal pouch 3 defect where the thymus itself is deficient in size and obviously will produce fewer numbers of T cells as a result. In addition, the paracortex region of the lymph node contains dendritic cells as well as high endothelial venules, which are sites where the lymph nodes are repopulated. Finally, in order to initiate an immune response, obviously there has to be an interaction basically between a B cell and a T cell and a corresponding antigen-presenting cell or a dendritic cell. And once the antigen-specific B and T cells pair, the B cells begin to divide rapidly, leading to the formation of the germinal center in the outer cortex and the subsequent proliferation and differentiation of subsequent B cells. And obviously dendritic cells and macrophages would be present in this area as well. Finally, let's look at the third of our lymphatic organs, again a secondary lymphatic organ, again a site of generation of immune responses, and this is the spleen. So certainly the spleen is involved in immunologic responses that are presented to it by the blood vascular system, but also a significant part of the spleen is involved in the recognition and removal of defective, misshaped, and or aged red blood cells and platelets. Functionally, the spleen certainly has an extensive blood supply provided by branches of the splenic artery that branch initially in the form of trabecular arteries, give rise to central artery branches, and subsequent branching pattern leading to the trabecular veins that ultimately will drain back into the splenic vein. And fundamentally, the two regions of the spleen are not well separated from one another, meaning that the spleen does not have a well-organized cortex versus a medulla. Rather, it is simply an integrated array of red pulp and white pulp. The white pulp, as the name suggests, consists of areas of B cells and T cells, usually in close association with the central artery. What we refer to as being the periarterial sheath is considered to be the T cell portion of the white pulp. The area of what's called peripheral white pulp contains the germinal centers that are highly populated mainly by B cells. The red pulp consists of what we call splenic cords of Billroth and venous sinusoids, and this, as we've alluded to earlier, is the site where defective red blood cells are filtered out of the bloodstream at this point. And also, this is going to be a site where defective red blood cells, as seen in cases of sickle cell anemia, hereditary spherocytosis, or thalassemia syndromes, are basically delayed in their passage from the open circulation back into the venous sinusoid and phagocytosed by macrophages that are lining these Billroth cords. In a diagram, we can kind of see a schematic illustration of where the regions of red and white pulp are found. Note the proximity to the central artery of the periarterial T cell rich sheath and the adjacent peripheral white pulp also associated in proximity with the central artery. And note interdigitating among that are trabeculae basically of red pulp, largely filling up the balance of the spleen. Now, in terms of vascularization, we know that most of the red pulp receives blood that's in what we call an open circulation, meaning that it's not bound by an endothelium and is basically outside largely the blood vascular system. The closed circulation of the spleen is a minor component of splenic vascular circulation, comprising only 10% of the vasculature. And basically, red blood cells must leave the spleen. In order to leave the spleen from the open circulation, red blood cells must pass through a mesh of reticular fibers and into the splenic sinusoids, basically by passing through a discontinuous basement membrane and a slit between these endothelial cells of that sinusoidal epithelium. And basically these red blood cells must negotiate this slit-like discontinuous basement membrane to leave the open circulation and get back into the venous sinusoids in order to exit from the spleen. The open circulation basically, therefore, allows red blood cells and blood vessels to open directly into the red pulp. And basically this path takes them from a trabecular artery to a central artery through a penicillar arteriole, basically exposing them to the macrophage and sheath capillaries in the red pulp. Closed circulation basically is a direct path, all endothelial line that simply allows a small percentage of the vasculature of the spleen to pass directly from a penicillar arteriole back into a splenic sinusoid. This summarizes our quick view of the histology of the different primary and secondary lymphatic organs, emphasizing again at histologic levels that the thymus and the lymph node have a distinct cortical and medullary regions. The spleen basically consists of areas of irregular red and white pulp, and secondarily the lymph node and the spleen also are characterized by the presence of germinal centers. Let's now move on to Chapter 5, which deals with the layers of stratified squamous epithelium that characterize the integument or skin. And first, let's begin by looking at the various layers of this stratified squamous epithelium, beginning at the most basal layer. This is known as the germinal or Malpighian layer. This is the layer that's situated immediately adjacent and lying on an underlying basement membrane. It consists mainly of columnar-like cells, basically that are regenerating all of the other cells in the stratified squamous epithelium. This, along with the spiny layer, which is immediately adjacent to it, basically are largely bound together by numerous desmosomal contacts, helping maintain the integrity of this epithelium, because certainly stratified squamous epithelial layers in skin are under considerable stress. Layer 3, as we move toward the surface of the skin, is the sealant layer. It's known as the stratum granulosum. It consists of more flattened epithelial cells filled with basophilic keratohyalin granules. And basically, when viewed at the electron microscope level, these are membrane-coated granules that provide the sealant necessary to protect this epithelium from penetration by substances outside the layers of the skin. As we move closer to the outside, a stratum lucidum is a transitional layer of epithelial cells that are basically losing or have lost their membrane and non-membrane bound organelles, so they're relatively clear staining and comprise pretty much a small single layer in the epidermis. But note that the stratum lucidum is only found typically in areas of the thickest skin, which is found in the palms of the hand and the soles of the feet. And finally, the outermost layer of the epidermis is basically the stratum corneum, which consists of very flattened anucleated squamate cornified keratinized cells that are basically close to being desquamated at the most superficial layers of the epidermis. The epidermis, fundamentally, all of these layers consists of various forms of keratinocytes. And basically these provide not only the dominant cell type in a stratified squamous epithelium of skin, but obviously the keratinocytes that are generating the keratohyalin granules provide the barrier function of the epidermis. Other specialized cell types that are found in the epidermis include melanocytes. These are derivatives of neural crests that migrate into the basal layers of the epidermis. Their job, as their name suggests, is to produce the pigment melanin in the form of melanosomes. Melanin is then transferred to the keratinocytes, and it really is the keratinocytes that convey the color to skin, even though the pigment melanin is produced in the much less numerous melanocytes in the basal layers. Some diseases associated with stratified squamous epithelium associated with skin, and it's important to make the distinction between pemphigus and pemphigoid. Pemphigus is an autoimmune blistering disorder caused by disruption of the desmosomes linking the keratinocytes in the basal layers of the epidermis. Psoriasis results from an increase in the number of proliferating cells in these basal layers, particularly in the basal plus in both the Malpighian layers, the stratum basale plus the stratum spinosum, resulting in basically a greater than normal epidermal thickness and a greater than normal turnover of those epidermal keratinocytes. Albinism occurs when melanocytes are actually unable to synthesize melanin. Vitiligo is a case where the melanocytes themselves are destroyed, And then generally this is secondary to an autoimmune related dysfunction. Both of these obviously would result in depigmentation, a lack of melanin pigments being taken up into the associated keratinocytes. Pemphigoid is a blistering disorder that occurs when hemidesmosomes are disrupted between the basal layer of the epidermis and the epidermal junction. So pemphigus was a desmosome disorder between keratinocytes. Pemphigoid is a blistering disease, again autoimmune, that basically results in a disruption of hemidesmosomes at the level of the dermis-epidermis junction. And then finally, let's look at a light micrograph of these various layers. You can see the pink layer at the bottom of this light micrograph is the underlying dermis, basically highly vascularized, containing nerve endings, specialized receptors, blood vessels, and so on and so forth. The actual epidermis itself is avascular, and you can see its stratified components here. The area indicated by letter B is basically the germinal layers, the Malpighian layers, the stratum basale as well as the stratum spinosum. The layer indicated by letter G is the stratum granulosum, the sealant layer. You can see a little more darkness, a little more basophilia associated with this area because of the keratohyalin granules. You can just make out a very thin, lucid layer that's situated immediately adjacent to the granulosum layer indicated by letter L. And then you can see the outermost cornified layer of stratified squamous cells having lost all of their organelles and close to being desquamated indicated by letter C. So this summarizes now Chapter 5, basically the fundamental elements of and function of some of the cell types associated with stratified squamous epithelium that's characteristic of the epidermis. Let's now turn our attention to Chapter 6, which deals with the histology and elements of the respiratory system. And on the first slide, we can basically summarize the bulk of the respiratory system by identifying the fact that it contains a large conducting portion whose job it is obviously to transmit inspired air down to the lungs, and along the way, basically, its job is to warm and moisten and filter the inspired air particularly during inspiration. The functional end of the respiratory system is certainly in the lungs themselves, dominated by the alveoli, where there is a mechanism, cellular and otherwise, to provide for the gas exchange between air and the underlying bloodstream. The nasal cavity kind of begins the area of the respiratory system. It's certainly dominated by a respiratory epithelium, and this respiratory epithelium is the classic pseudostratified ciliated columnar epithelium intermingled with goblet cells, underlain by a combination of mixed mucus and serous glands. And basically this respiratory area that largely dominates the nasal cavities, whose job it is to warm, moisten, and filter inspired air. The adjacent olfactory area, a small part of the nasal cavity in the upper part of the nasal cavity, basically is also lined by a pseudostratified epithelium, but it largely consists of bipolar olfactory neurons, their adjacent supporting cells, brush cells, and basal cells. The basal cells, their job is to act as stem cells to continually replace the olfactory receptor cells, because the olfactory receptor cells are really neurons and are really the only example in the adult central nervous system where neurons are continuously replaced. Underlying this epithelium, basically the underlying Bowman's glands produce serid fluid, which basically allows the odorous substances to be dissolved in fluid. Now let's look at the overall additional histologic features of the respiratory portion, or the conducting portion of the respiratory system. It began in the nasal cavity with a pseudostratified ciliated columnar epithelium, basically associated with goblet cells, and this continues down into the trachea. The trachea, as we know, is supported and lined by an array of C-shaped cartilaginous rings, giving it a very distinct feature in imaging, underlain by seromucous glands, and with a small opening at the open ends posteriorly of the C-shaped cartilages lined by smooth muscle. As we move down into the primary and secondary bronchioles, we undergo a transition from a pseudostratified ciliated columnar epithelium to a more simple columnar epithelium. The cartilaginous rings of the trachea are gradually replaced by irregular plates of cartilage at the level of the bronchi. There are fewer glands, but the smooth muscle tends to be more prominent. As we move down to the conduction bronchioles, basically we now have made the transition to a more columnar epithelium, still ciliated, intermingled with goblet cells, but now we find the more distinct Clara cells down at the more distal level of these conduction and ultimately into the respiratory bronchioles. No longer do we have cartilage, and basically the glands have largely disappeared as well. But as we have moved from the trachea to the bronchi to the bronchioles, both the smooth muscle and the elastic fibers have become much more prominent. Clara cells in recent years have gotten a lot of attention in terms of their functional significance, and as we note at the bottom of this chart, Clara cells basically, in addition to type 2 pneumocytes, produce a form of surfactant that reduces alveolar surface tension. And more recently, we know that Clara cells basically have a chloride transporter that's basically involved in the transport of chloride out of Clara cells into the underlying, or the lumen of the respiratory epithelium. Finally, if we move down to the level of the alveoli themselves, we can see the distinct structure as illustrated in this diagrammatic view. We know that 90% of the alveolar luminal surface is lined by the squamous type 1 pneumocytes that basically are a major element of the blood-gas barrier. And you can see down at the bottom how those type 1 pneumocytes, their thin cytoplasmic processes, share a common basal lamina with the underlying endothelial cells that line the pulmonary capillary blood cells. In addition, about 10% or 5 to 10% of the luminal surface of an alveolus is lined by the cuboidal type 2 pneumocytes, which we know their claim to fame is that they are the surfactant producing cells that basically line the alveolus and reduce alveolar surface tension. And we know functionally in fetuses that surfactant production begins after week 25 of fetal life. And in addition, inside the lumen of the alveolus, we find alveolar macrophages. Their job is to trap basically airborne particles, as well as clear basically surfactant from the luminal surface of the epithelium. Finally, in terms of generation, type 1 pneumocytes, once generated, are postmitotic. So it is left up to the type 2 pneumocytes to generate both new type 2 cells as well as new type 1 cells, respectively. Finally, just in summary, we made this comment already, the type 1 pneumocytes basically cover virtually all of the luminal surface epithelium, 95% or so, and provide a thin squamate cytoplasmic surface that's associated with gas exchange. And as we know that they are a major component on the lung side of the blood-gas barrier, and basically oxygen that's delivered by the respiratory system to the level of the alveoli is allowed to pass through the cytoplasmic processes of the type 1 pneumocytes through a shared basal lamina and into the capillary lumen after passing through the cytoplasm of the capillary endothelial cell. Type 2 cells, as we indicated, are cuboidal, very distinct in the electron microscope, not only because of their cuboidal shape, but also because of the membrane-bound whorls or lamellar bodies that basically indicate the presence of the surfactant, which is obviously the major secretory product of these cells that's released into the lumen of the alveolus to help reduce alveolar surface tension. This now summarizes basically the elements of Chapter 6, which dealt with the fundamental cytological features of the respiratory system. Certainly, the bulk of the respiratory system is involved in conduction of inspired air down into the lungs. Once we reach the alveolar level, be sure to recognize the difference between a type 1 pneumocyte, a type 2 pneumocyte, and the functional significance of Clara cells versus alveolar macrophages. Let's now visit the elements of the gastrointestinal system, with a commentary on its major accessory gland, that's of the liver, with a view at the elements illustrated in Chapter 7. Basically, we're talking about an extensive system that consists mainly of the epithelium and underlying structures of the digestive tract, as well as its associated glands. The gland we'll concentrate the most on, obviously, is going to be the liver. First off, let's illustrate in 2 slides, one that's in the notes, one that we incorporated into this video, that illustrates the fundamental histologic similarities in terms of layers of the digestive tract. Innermost and closely associated with the lumen of different components of the digestive tract is the so-called mucosa, and the mucosa of the GI tract consists of a lining of epithelium, which we'll see is mostly going to be a simple columnar epithelium, an underlying vascular and connective tissue, lamina propria, and immediately deep to that will be a layer of muscle known as the muscularis mucosa. It is in the area of the mucosa where we see the most distinct differences in various components of the GI tract. Underlying the mucosa will be an area of submucosa, then in one region of the GI tract, the duodenum, which will contain some prominent glands. Underneath that are circular and longitudinal layers of the muscularis externa, basically consisting of smooth muscle. And in various parts of the GI tract, there may be a serosa or peritoneal covering, either completely or virtually completely covering a layer or a portion of the GI tract that's peritoneal, or only partially covering a portion of the GI tract if it is secondarily retroperitoneal. So now a better picture that we borrowed from Kierszenbaum's histology and cell biology text basically serves to illustrate all of the different structural features in one picture of the regions of the GI tract, beginning with the esophagus and going down to the area of the large bowel of the colon. And again, you can see, as illustrated here, and then we'll underscore this in chart form, the major histologic differences in regions of the GI tract come in different parts of the mucosa. And again, you can see fundamentally, as we illustrated previously, the structural layers of the GI tract begin with a mucosa, which is epithelium plus underlying lamina propria, plus a thin layer of the muscularis mucosae. Underneath that, a submucosal layer that may or may not have some glands in it. And then the muscularis layer consisting of longitudinal and circular layers of smooth muscle. And then finally, an outer layer of adventitia, again connective tissue, that may or may not be in close contact with an overlying layer of serosa, which is the peritoneum. So let's go through these histologic layers and differences, and then underscore them with the specific cell type features that are in the charts on the subsequent couple of pages. First off, note that the esophagus is distinct in terms of its epithelium because it's the only part of the endoderm lining of the gut tube that's characterized by a stratified squamous, non-keratinizing epithelium. Basically, other than that, the histology of the esophagus is unremarkable except for the presence of some underlying submucosal glands that open periodically into the lumen of the esophagus. Because in a way, certainly the esophagus is merely a conveyance tube to get the bolus of food from the pharynx down to the stomach. The stomach epithelium begins our tour of epithelium that's basically thrown into pits and deep and shallow gastric glands, all lined by different functional elements of a simple columnar epithelium. Other than that, the underlying lamina propria and muscularis mucosa layer are unremarkable. Once we get down to the duodenum, the simple columnar epithelium is largely absorptive, highly microvilliated, and throughout the length of the small intestine, duodenum, jejunum, and ileum, the lamina propria and the overlying epithelium are thrown into distinct finger-like projections known as villi, which certainly serve to increase the surface area available for absorption of these 3 regions of the small intestine. In the duodenum, we'll be able to distinguish a duodenum section not by its epithelial and underlying lamina propria characteristics, because all parts of the large and small intestine not only have a simple columnar epithelium, but as you can see, all parts of the small and large intestine are characterized by the pronounced invaginations known as crypts of Lieberkühn. They're not actually labeled on this. Oh yeah, they are labeled on this particular diagram, as illustrated by those white lines projecting down deeply into the lamina propria. The duodenum is distinct from jejunum and ileum by the presence of the submucosal Brunner's glands, which are producing an alkaline secretory product to counteract the acidic medium that's being presented to the duodenum from the lumen of the stomach. The jejunum and ileum certainly are going to lack the submucosal Brunner's glands, but the way you can distinguish histologically the jejunum from the ileum is by the presence of prominent patches of B lymphocytes in a Peyer's patch situated immediately adjacent to the luminal surface in the underlying lamina propria. So the fundamental distinctions between areas of the small intestine are this. The epithelium is simple columnar absorptive thrown into folds or villi, and basically crypts of Lieberkühn characterize all parts of the small intestine. But in the duodenum, the submucosal area will have Brunner's glands. In the ileum, the underlying lamina propria will be characterized as by Peyer's patches. In the absence of both, obviously the indications are that you are in a section through the jejunum. The colon is still going to have the simple columnar epithelium, but it's only going to have the crypts of Lieberkühn because the villi will now be basically absent. So now in chart form on the next page, we can summarize these differences with a kind of a look at some of the specialized cell types that are found in various areas of these regions of the gut tube. Certainly, as we've indicated, the esophagus is the only part of the endoderm lining of the gut tube that has a non-keratinizing stratified squamous epithelium, which we've underlined as indicated here. Differences in the muscularis portion of the esophagus is the transition that takes place from skeletal muscle in the upper third to completely consisting of smooth muscle in the lower third. Basically, the middle third of the esophagus probably has a mix of both skeletal and smooth muscle. The skeletal muscle in the upper portion of the esophagus is being innervated by basically skeletal motor neurons in the vagus nerve, whereas the skeletal muscle in the lower portion of the esophagus is innervated by autonomic nerve fibers, also carried by the vagus nerve, which obviously answers the innervation question that's raised here at the top of this chart. By the time we get to the stomach, basically now the major characteristics of the epithelium are simple columnar. The body and the fundus of the stomach are thrown into shallow pits and deep gastric glands. The characteristic cell types are numerous in the body and the fundus of the stomach, consisting of, well, a variety of mucus cells that serve to protect the corresponding epithelium from the acidic medium that's in the lumen of the stomach. Also found at various points down at these in these deep gastric glands are chief cells that produce pepsinogen and lipase precursor. They tend to be found at the bottom of these glands. Parietal cells, a variety of locations, both near the luminal surface and more deeply placed. Parietal cells, their major job certainly is to produce hydrochloric acid, but their other major secretory product, which is essential for life, which enhances, obviously, or enables the absorption of vitamin B12 from the terminal part of the ileum, is intrinsic factor. And then certainly there are going to be a variety of enteroendocrine cells that are populating these gastric pits. As we move down to the pyloric portion, we basically have the same array of cell types, but most importantly, this is where we find a concentration of enteroendocrine cells that are producing gastrin. Gastrin's major role is to induce parietal cells to increase their secretory output of hydrochloric acid. As we move now down to the portions of the small and large intestine, as we alluded to earlier, we certainly can say that a common feature of all parts of the small intestine, duodenum, jejunum, and ileum, is the presence of the absorptive simple columnar epithelium, but basically the characteristic features are that all parts of the small intestine are thrown into prominent villi, folds, and interdigitated with crypts of Lieberkühn. So we have to use other criteria histologically to distinguish a section of duodenum from jejunum and ileum. And as we allude to here, The duodenum basically can be distinguished by the presence of the submucosal Brunner's glands, which, as we alluded to, are producing an alkaline secretion to counteract the acidic medium presented to it from the stomach. And the jejunum, basically also characterized by the prominent villi, crypts of Lieberkühn, and so on and so forth. But the ileum can be distinguished from the jejunum by the presence of the patches of lymphatic tissue in the form of Peyer's patch nodules immediately adjacent to the luminal surface epithelium. Down in the crypts of Lieberkühn, in all parts of the small and large intestine are the Paneth cells. Their job is to produce lysozyme, which has a bacterial regulating role. Goblet cells are intermingled among the columnar absorptive cells throughout the length. As we saw in the stomach, the functional significance of the enteroendocrine cells changes, whereby in the duodenum, the EE cells are producing mainly cholecystokinin to basically stimulate the release of bile from the bile duct and the gallbladder, and to basically stimulate the release of secretin as well that increases the output of bicarbonates from the exocrine pancreas. The jejunum, again, same cell types except for differences in the EE cells in the overlying epithelium. The ileum, as we said, has the Peyer's patches, and basically overlying the Peyer's patches are squamate or slightly dome-shaped M cells or microfold cells. These are antigen-presenting cells that basically take up and transport antigen from the lumen of the ileum and present it to the underlying B cells that will generate germinal centers in the underlying Peyer's patches. Large intestine, same basically histological features in terms of its epithelium, but now we lack the villi that were prominent features of all regions of the small intestine. The large intestine still has the crypts of Lieberkühn, and obviously the simple columnar epithelial cells are still mucosecreting and absorptive, but now the absorption basically is involving the active transport of sodium out of the lumen of the large intestine, as well as the passive movement of water down its concentration gradient. Histologic features, now we're looking at a diagrammatic feature of a villus, and you can see its overall lining of enterocytes, the columnar absorptive epithelial cells intermingled with goblet cells. Down at the base of a crypt of Lieberkühn is basically where we find the stem cells that are involved in the regeneration of enterocytes throughout the length of the villus. And also down in the bottom of the crypts of Lieberkühn are the presence of the Paneth cells, which produce their secretory product in the form of lysosome. Finally, in terms of histology, this picture is a distinct view of the section through the duodenum, and the reason we know that, certainly we can see the viliated columnar epithelium with the intermingled with the crypts of Lieberkühn, but what tells us this is a duodenum section is certainly by the presence of the prominent submucosal Brunner's glands. And again, we raised the point about what's innervating the smooth muscle down here that in this, both the longitudinal and circular layers, and the innervation is provided by a myenteric plexus, and again, as we saw in smooth muscle in the esophagus, the entire foregut, including the portion of the duodenum up to the entrance of the common bile duct, is being innervated parasympathetically by the fibers in the parasympathetic axons in the vagus nerve. This innervation also will carry out through the length of the midgut as well, basically the changeover occurring 2/3 of the way across the transverse colon, where the parasympathetic innervation of smooth muscle will be taken over by pelvic splanchnic nerves, sacral outflow of S234. Let's move down to an actual histologic section. We can see again quite dramatically a view of the duodenum. You can see again the villi and crypts of Lieberkühn, but the underlying submucosal glands of Brunner give this away as being a prominent section through the duodenum. Let's move now to a section in the notes that illustrates a section through the ileum, and the reason we know it's ileum is not again by the presence of the villi and the crypts, but by the presence of prominent patches of lymphatic tissue containing B cells and the related germinal centers in close proximity to the luminal epithelium. And even though we can't distinguish it histologically, that luminal epithelium is characterized by the presence of the microfold or dome cells that are the antigen-presenting cells that are kind of intermingled among the adjacent columnar enterocytes at the level of the Peyer's patches. In our next slide, let's take a view of and talk about the functional organization of the liver. And fundamentally, we're looking now at a section through a liver lobule. Basically, the organization that we want to talk about is the differences in the direction of blood versus bile flow through a hexagonal liver lobule. First off, at each of the 6 points of the hexagon of this liver lobule is what we call a portal triad, whereby there are 3 tubular structures. 2 of them are vascular. 1 of them basically are receiving all the tributaries of bile canaliculi, delivering bile to the perimeter of each point of the lobule. So the 2 vascular structures at each of the portal triads are a tributary, a branch of the hepatic portal vein, and obviously a branch of the hepatic artery delivering either poorly oxygenated, nutrient-rich blood to the liver or oxygenated blood to the liver in the form of coming from the hepatic artery. Certainly the portal vein is delivering the dominant source of blood supply to the liver in the range of about 70% of liver blood supply is coming through that vascular structure. The remaining 30%, in this case of arterial blood, is being delivered to the liver by way of the hepatic artery. Note that blood is flowing from each of the perimeter points of the hexagon toward the central vein, which is basically draining the blood out of each of the liver lobules and is receiving blood, obviously, from each of the 6 points of the hexagon. So blood is flowing from the perimeter of the hexagon toward the central vein in the direction of the blue arrow. In contrast, bile that's being produced by liver hepatocytes and is basically being collected in bile canaliculi between adjacent hepatocytes is basically being collected and delivered toward the perimeter of each point of the hexagon. So the point is, bile is flowing in a direction that is opposite to that of blood flow. So in the inset diagram on the right side of this page, you can see a liver sinusoid, basically that's lined by a discontinuous sinusoidal endothelium. Inside the lumen of that sinusoid, we find the Kupffer cells, the macrophages associated with the liver. And between those sinusoidal endothelial cells and the luminal surface of each of the plates of hepatocytes is the space of Dease. The space of Dease is basically where all of the endocrine-based products of a hepatocyte are released into the bloodstream. They would be albumin, prothrombin, and so on and so forth, proteins. Whereas you can see the location of bile canaliculi not associated at all with the liver sinusoids. The canaliculi, as you can see, are associated with small channels between the adjacent hepatocytes into which bile is secreted. And certainly if bile is being secreted and moving in the direction of the black arrow, as we've indicated, that blood flow would be moving in the opposite direction. So, liver hepatocytes basically are the marginal functional cell type that is associated with the liver. These are highly regenerative cells. About as 90% of them can be replaced in a very short period of time, basically as little as 2 weeks. These are linked together by prominent series of gap junctions and basically separated in part by the bile canaliculi that is the site where basically the exocrine product, bile, is being produced. We've already pointed out that the microvilliated free surface of the hepatocytes are exposed to the space of Dease, and it is into the space of Dease that the so-called endocrine products of a liver hepatocyte, mainly in the form of large proteins, are released into the bloodstream. Basically, hepatocytes have abundant glycogen that's being delivered to them from the portal system that's been absorbed out of the bloodstream, prominent mitochondria, that are characteristic typically of a liver cell. And as we've indicated, the major proteins bound for export into the bloodstream are large proteins, albumin, prothrombin, fibrinogen, so on and so forth. Those are the endocrine products. The exocrine product, certainly of the liver, is associated with bile, and certainly a complete course in biochemistry could be dominated alone by what some of the vast functions of the hepatocyte are. The liver, as we said, basically contains the sinusoids that are lined by a fenestrated capillary endothelium. Inside that endothelium, inside the lumen of the sinusoid, are the phagocytic Kupffer cells, which are part of the whole overall mononuclear phagocyte system. Their job overall is to phagocytize red blood cells. So in essence, if a patient has undergone a spleen removal because of hereditary spherocytosis, the liver Kupffer cells can kind of take over many of the functions of the red pulp associated with a defective spleen. A key cell type that is found in the space of Dease are the Ito cells, or the lipocytes. Their job basically is to store fats and vitamin A, but as we see in patients that develop cirrhosis of the liver, basically late-stage manifestation of chronic alcohol abuse will cause those lipocytes to begin to secrete collagen, and that collagen basically ultimately will block up the space of Dease and the liver sinusoids and alter portal venous blood flow through those sinusoids. Now, on this chart, on this page, we can illustrate basically another feature about how the liver is organized. Since we know that the liver lobule is organized into a blood and bile flow diagram, we basically can subdivide the regions of a liver lobule into zones. Zone 1, as illustrated here, is closest to the point of entry of blood to the perimeter of the lobule. Zone 2 is intermediate. Zone 3 is furthest away from the entry point of blood and is certainly closest to the central vein. So functionally, we can associate different parts of the liver according to these zones as being first or last affected by either drugs or anoxia, respectively. For example, patients that are associated with chronic alcohol abuse tend to develop signs of a fatty liver, and this basically characteristically affects zone 3 first, as well as certainly anoxia, sudden decrease in arterial blood supply of the liver, will affect the zone of the liver furthest away from the entering blood, and that's also going to be zone 3. Certainly drugs are going to affect hepatocytes in zone 1 first, because they're going to be the first region of hepatocytes that are going to be affected by those entering drugs. And then finally, let's look briefly at the associated glands that are part of the gastrointestinal system. We know that there are 3 different kinds of salivary glands: parotid, submandibular, and sublingual, having different levels of serous versus mucus types of activity. The parotid gland, high amylase activity, is a pure serous gland, whereas the submandibular and sublingual glands are mixed glands, submandibular being mainly serous, sublingual gland being mainly mucus. And we know that their cranial nerve secretory secretomotor innervation is slightly different. The parotid gland is under secretomotor control by parasympathetic axons in the glossopharyngeal nerve, whereas the submandibular and sublingual glands are under parasympathetic innervation by cranial nerve 7. This summarizes our brief tour of the different histological features of not only the gut tube in terms of its epithelial differences, underlying lamina propria and submucosal differences, but we've also looked briefly at the histological features of the liver, liver hepatocytes, and the importance of Kupffer cells as well as the perisinusoidal lipocytes. And we ended finally with a brief look at a reminder of the different histological features of the salivary glands. Let's now turn our attention to our next section, and this is Chapter 8 that involves the histologic elements of the urinary system. In our first picture here, we simply illustrate the fundamental organization of the kidney, and note that it's divided mainly into a cortex and medulla, with basically a hilum that faces medially. The hilum is basically the point of entrance and exit for the vasculature, renal artery and the renal vein, and the point of origin of the ureter. The renal pelvis, which is the expanded upper portion of the ureter, actually becomes subdivided as we moved into the area of the kidney into 2 or 3 major calyces that then subsequently receive several up to 8 minor calyces. And as we'll see, each of those minor calyces is associated with the apex of a medullary pyramid. And certainly branches of the renal artery vein and its nerve supply basically supplies all parts of the corresponding kidney. Pictorially, we can see the arrangement of the area of the hilum first, which, as illustrated on here, is the point of exit of the renal pelvis, the dilated upper portion of the ureter, which is basically a tube of smooth muscle. And you can see how the major calyces, 3 or 4 of them, contribute to form the renal pelvis, and how up to 8 minor calyces join together to form the series of major calyces, again contributing to the drainage of the kidney. And you can also see the apex of the medullary or renal pyramids, basically which are the site where the collecting ducts enter or exit and drain their corresponding urine into the minor calyces. The cortex, you can see, is outermost, containing obviously major components of the uriniferous tubule, the nephron, but notice we'll see that parts of a nephron are going to extend down into the renal columns between the medullary or the renal pyramids. Being more specific, we know that the cortex basically contains the major components of the uriniferous tubules, and the medulla basically consists of an array of medullary pyramids, largely containing collecting ducts, but separating those medullary pyramids will be an array of renal columns containing basically the loops of Henle of a corresponding series of uriniferous tubules. The actual nephron consists of a renal corpuscle, which is a glomerulus, a tuft of capillaries surrounded by Bowman's capsule. Then from the renal corpuscle in Bowman's capsule emanates the proximal convoluted tubule, the loop of Henle, which we can see extends down into the renal columns in the medulla. And then basically the ascending limb becomes continuous with the distal convoluted tubule. When the nephron joins with the collecting duct, that basically forms the so-called uriniferous tubule. The collecting ducts then, as we illustrate in this picture, open into one of the 8 or so minor calyces. Let's look first at the renal corpuscle, which we know consists of a tuft of arterioles provided by or containing blood from an afferent and efferent blood vessel surrounded by the elements of Bowman's capsule. This obviously is where we find the filtration barrier associated with the kidney, such that the afferent arteriole enters, providing arterial blood to the kidney, and basically it's covered by what we call the visceral layer of Bowman's capsule, which is characterized by the distinct podocytes in their foot processes. The parietal layer is continuous with the visceral layer, is outermost, and between the parietal and visceral layers of Bowman's capsule is the so-called urinary space, into which we will find the ultrafiltrated blood plasma after it's been filtered through the corresponding filtration barrier. The inset area of detail in this picture illustrates at least diagrammatically, the elements of the so-called filtration barrier. Basically, we can see a fenestrated capillary endothelium lining the afferent and efferent, the capillary bed interposed between the afferent and efferent arterioles. A shared basal lamina, and on the other side of the basal lamina we find the foot processes separated by small diaphragms of the podocytes, and then on the other side of the cytoplasm of the podocytes we find the so-called urinary space interposed between the parietal and visceral layers, the podocyte layer of Bowman's capsule. In an electron micrograph on the next slide, first at low magnification on the left, we can see an illustration of a number of podocyte nuclei and their adjacent cytoplasm, and you can see many examples of the podocyte foot processes extending down to become in direct contact with the shared basal lamina between the podocyte foot processes and the discontinuous fenestrated capillary endothelium. At higher magnification on the right side of this picture, again, podocyte cytoplasm is evident. You can see some cytoplasmic processes of that podocyte emanating down to form part of the filtration barrier. The area indicated by letter E is the lumen of the fenestrated capillary endothelium. The area indicated by B on the other side of the podocyte foot processes is the urinary space. And certainly, as we've described earlier, the major elements of a filtration barrier such as this one is the presence of a type 4 filtration collagen that characterizes the basal lamina. And as you'll hear about in pathology, much of kidney pathology is caused by changes in the basal lamina, particularly in cases of Goodpasture syndrome where antibodies are raised against the type 4 collagen in the basal lamina, or in patients that develop Alport syndrome where basically there's a splitting or thickening of the basal lamina that is similar in a disease process to one that also affects the stria vascularis, which is the site of where endolymph is produced in the inner ear. Now let's kind of summarize functionally what the different components of the nephron distal to Bowman's capsule are involved in, beginning with the proximal convoluted tubule. As indicated here, it's certainly the longest, most convoluted segment of the nephron, and basically it's where much of the glomerular filtrate is resorbed back into the proximal tubule. And basically, this has a dramatic surface area characterized by a tremendous brush border and a large surface area specialized for resorption and secretion, respectively. As we move down into the loop of Henle, the loop of Henle forms a hairpin loop of the nephron that extends down into the medulla and consists of thin and thick segments. But the descending and ascending limb have distinct differences relative to their permeability to water. The descending limb is permeable to water and less so to urea and sodium chloride, such that the fluid at the bend of the loop of the Henle, deep in the medulla of the renal column, is hyperosmotic relative to blood plasma. The ascending limb is impermeable to water, but permeable to sodium chloride and urea. So therefore, by the time the fluid reaches the distal tubule, it is now hypoosmotic relative to blood plasma. In the distal convoluted tubule, it's lined by a more cuboidal epithelium whose major job is to resorb sodium chloride, but now not urea, from the tubular filtrate. By the time we reach the collecting tubules, basically these are the part of the nephron that are basically under control or respond to antidiuretic hormone or vasopressin, that's the posterior aspect of the pituitary, and vasopressin regulates the permeability of the collecting tubules to water. So therefore, the collecting tubules become kind of the final common denominator in regulating water conservation and the concentration of the resultant urine. Finally, let's look at some specialized cell types that are associated, again, back at the level of the renal corpuscle. These are mesangial cells that are either intra or extraglomerular. What's illustrated here are some extraglomerular mesangial cells, and some specialized cell types that are known as juxtaglomerular cells that are basically lining the lumen of the afferent arteriole, and the adjacent macula densa cells, which are taller, more columnar cells in a closely associated wall of the distal tubule. Functionally, the juxtaglomerular cells are basically modified myoepithelial cells, which we've seen in the wall of the afferent arteriole. Their job is to secrete renin, which basically converts circulating polypeptide angiotensinogen into angiotensin 1. And then subsequently, AT1 is converted to AT2, which is a potent vasoconstrictor that stimulates aldosterone secretion from the adrenal cortex. And certainly, as we know from physiology, aldosterone increases sodium and water resorption in the distal portion of the nephron. The macula densa cells, as we indicated, are taller, more columnar cells in the adjacent distal tubule, and basically their function is thought to sense the sodium concentration in the area of tubular fluid. So now, this is a change in the notes. We identified these cells in the notes as being Pulkesin cells, but now we know much more about them than was originally stated in the notes. These extra and intraglomerular mesangial cells are actually parasites, which in our case, as we illustrated previously, are found between the afferent and the efferent arterioles near the vascular pole of the glomerulus. These cells we now know are acting as parasites are contractile, phagocytic, they are capable of synthesizing collagen, and they produce factors that induce constriction of the glomerular arterioles and therefore can regulate the filtration rate by changing the luminal diameter of those arterioles. So they have much more of a functional significance than we previously thought. Finally, a comment, and I've added this as well, about erythropoietin. Erythropoietin is the major regulator of erythropoiesis, logically, and it is a glycoprotein that's produced mainly in the kidney. And what can come up on the exam is what kind of cells and where is this produced? As we know now, it's produced by what are known as peritubular interstitial cells that are largely found in the inner part of the cortex of the kidney and in the outer part of the medulla. This kind of summarizes our functional histologic view of the elements of kidney tissue, beginning with a look at the renal corpuscle, its elements of the filtration barrier, podocytes, fenestrated capillary endothelium, and the functional significance of its united basal lamina, as well as a brief look at the functional differences between the proximal and distal tubules and the intervening elements of the loop of Henle. Finally, we illustrated just the functional significance of the juxtaglomerular apparatus, consisting of JG cells modified in the wall of the afferent arteriole, the adjacent macula densa cells that sense sodium concentration in tubular fluid, modified cells in the distal tubule, as well as a new look at the functional significance of the mesangial cells, as well as the probable location of cells that produce erythropoietin. Let's now turn our attention to the male and female reproductive system histology, beginning with a view of the male reproductive system illustrated in Chapter 9. First off, let's look at the functional end of the male reproductive system by looking at the various cell types and differences in a section through a seminiferous tubule. This is certainly the site in the male reproductive system where gametogenesis is occurring, beginning basically at puberty and progressing in waves throughout basically teenage and adult life. But recognize that the major functional cell type that extends both from the basement membrane to the luminal surface of the seminiferous tubule is that of a Sertoli cell. The Sertoli cells are basically the support cells that maintain and protect the developing gametes that are basically in various stages of gametogenesis. Note that, as we've noted on the slide, that Sertoli cells also secrete and produce Müllerian inhibitory substance, whose job it is during fetal life to suppress the elements of female reproductive structures. So the point we want to make on here is, first off, what is the time course and where do we find various gametes in various stages of gametogenesis? First off, you can see situated closest to the basement membrane the different stages of type A and type B spermatogonia, which basically are undergoing mitotic division in the early stages of gametogenesis. These two cell types are basically outside the blood-testis barrier. The blood-testis barrier is maintained by tight junctions between adjacent Sertoli cells. So the answer to the question of what the blood-testis barrier protects, it basically protects all of the gametes that are in various stages of meiosis I and meiosis II, beginning with the primary spermatocytes that are undergoing meiosis I. The spermatogonia, on the other hand, are not protected by the blood-testis barrier. So now, in answer to the other question, and this goes back to fundamentals of meiosis, is how many sperm cells are formed from a primary spermatocyte? And the answer is 4, because a typical primary spermatocyte will subdivide equally into a secondary spermatocyte where the chromosome number is reduced from 46 to 23. A secondary spermatocyte will proceed through meiosis II and form haploid spermatids that now have a complement of 23 chromosomes and a complement of 1N DNA. It is the spermatids that then undergo spermiogenesis to basically form mature spermatozoa, an example of which you could see basically with tails emanating out into the luminal surface of the seminiferous tubule. So the other significant cell type that's characteristic of a seminiferous tubule is found beneath the basement membrane, and this is the location of Leydig cells, which are a major testosterone-producing cell associated with the male reproductive system. And finally, to address the question that we'll compare when we talk about the female reproductive system, and that is, what kind of germ cells are present in the testis at birth? Well, since no stage of gametogenesis has begun at birth, basically in the testis at birth are nothing more than primordial germ cells that won't begin any stage of mitosis or meiosis until the individual reaches puberty. Next, let's look at the anatomy of a mature sperm cell itself that's completed the stages of spermato as well as spermiogenesis. And fundamentally, we can divide a mature sperm cell into a head, a middle piece, and a principal piece that largely forms the tail. The head contains, obviously, the genetic material, a complement of 23 chromosomes and 1N DNA. The head is capped by the acrosome. Remember that the acrosome is derived from the Golgi apparatus, and it contains the hydrolytic enzymes which will enable the head of the sperm cell to penetrate the zona pellucida, a necessary event if fertilization is to occur. And once fertilization occurs, we know that cortical granules are released from the oocyte that prevents additional sperm penetration. The middle piece of the sperm cell is the energy source because it contains the mitochondria that generate the ATPase motor activity that basically helps convey motility to the ciliated flagellum. And sperm, basically, once they enter the female reproductive tract, are viable for a period of up to 72 hours. Now let's look at the genital ducts that basically connect the seminiferous tubule ultimately with the outside world. Fundamentally, the mature sperm cells, once they leave the seminiferous tubule, enter what is known as the reedy testis, an array of tubules that basically connect the seminiferous tubules, move the gametes out of those tubules, and basically go from the tubula recti, the straight tubules, into the adjacent reedy testis. Then from the reedy testis, those ductules are drained by the efferent ductules. These are defined histologically by a pseudostratified ciliated epithelium. From the efferent ductules, sperm are moved to the epididymis, which is a long tortuous duct that begins basically at the head of the sperm, goes at the head of the testis, goes down to the tail of the epididymis where it becomes continuous with the ductus deferens. This has kind of a unique, histologically distinct epithelium in that it's still pseudostratified, but basically its luminal surface is dominated by the presence of stereocilia rather than true cilia. Basically, this is a major site where sperm undergo maturation, increasing their motility and their fertilizing capacity. The ductus epididymis is continuous with the ductus deferens, which is basically a smooth muscle tube which passes through the inguinal canal into the male pelvis and dilates gradually as it approaches the upper border of the prostate into a dilated or ampulla. And fundamentally, when the distal portion, the ampulla of the ductus deferens, joins with the duct of the seminal vesicle, basically what is formed is the ejaculatory duct, which then penetrates the prostate and empties not only the seminal vesicle secretory products, but also the sperm into the prostatic urethra. Seminal vesicles themselves secrete an alkaline, slightly viscous fluid that is rich in fructose that contributes an energy source for the sperm and, as well known, is not a storage site for sperm cells despite the name. The prostate, on the other hand, also contributes a secretory product that contributes to seminal fluid. It's largely rich in acid phosphatase, zinc, lipids, and citric acid, and often contains various concretions, particularly in humans, that are comprised of protein and carbohydrates. The prostate obviously has dramatic clinical significance because it's a major site of either benign or metastatic enlargements. Typically, male patients that develop benign prostatic hyperplasia, that enlargement typically develops in the periurethral zone. The metastatic adenocarcinomas tend to develop in the more peripheral parts of the gland, and if they are metastatic, classically prostate adenocarcinomas are well known as their site of metastasis basically is delivered to bone, particularly the vertebrae. And just a fundamental distinction to be made about lymphatic drainage associated with the testis. Testicular lymphatic drainage basically passes back along the length of the ductus deferens through the inguinal canal and basically is a direct lymphatic pathway that takes a testicular carcinoma, if metastatic, back to para-aortic or lumbar nodes. Scrotal drainage, on the other hand, basically passes initially to superficial inguinal nodes, which would not be involved in a patient with a metastatic testicular carcinoma. This kind of summarizes our brief view of the elements of the male reproductive system, beginning with the different stages of gametogenesis, the location of the so-called blood-testis barrier and which gametes are protected by it, and then just an array of the ductular structures that convey sperm from the seminiferous tubules to the outside world, with an additional comment on some of the anatomic features of the prostate. Let's finish our survey of histologic features of organ systems by looking at the elements of the female reproductive system that are illustrated in Chapter 10. First off, let's make an overall view of the elements of the female reproductive system. It's certainly going to begin with the business end of the female reproductive system, and that's where oogenesis is occurring at the level of the ovaries. But then the ovulated oocyte will pass through a discontinuous duct system, including elements of the uterine or of the fallopian tubes, the uterus down through its neck, the cervix, vagina, and basically out through the vestibule associated with the external genitalia. The business end of the female reproductive tract are the ovaries, and basically oogenesis occurs in the area of the cortex, closest to the outer surface of the ovary. The medulla, on the other hand, basically contains the blood vasculature. When we actually look at the structure of the ovary, we can see in this illustration different stages of ovarian development. But we want to make the point that basically oogenesis begins in utero, and basically oogenesis not only begins in utero, but at birth, all of the oocytes are basically primary oocytes that are arrested in the prophase stage of meiosis I. And they're going to remain in prophase basically from the time of birth until basically follicular and oocyte development resumes at levels of puberty. So obviously the answer to the question of what's in the ovary at birth is basically all primary oocytes arrested in the prophase stage of meiosis I. So this is the point that the ovarian follicles are situated in the area of the cortex, and the oocytes themselves are found by surrounded by a secretory or follicular epithelium that is formed by either follicular or also known as granulosa cells. And basically, at birth, there are 400,000 primary follicles, but as we know, only a very small percentage of those 400,000 primary follicles will actually reach maturity and the adult and will be subsequently ovulated at the rate of 1 a month. The remaining follicles basically are degenerating progressively through atresia, and atresia can occur at any stage of follicular development, particularly in post-adolescent life. Secondary follicles, which basically begin their development at puberty, where the primary follicle completes meiosis I and begins to go through the stages of meiosis II, basically are characterized by the presence of a follicular cavity, a surrounding area of cumulus cells, a corona radiata, and basically an adjacent connective tissue area that contains the theca interna and external cell layers. The theca interna, basically outside the basement membrane that incorporates the oocyte, produces androgens that are then converted into estradiol by the granulosa cells that are basically inside the corresponding basement membrane. When the secondary follicle develops, what addition forms around that secondary follicle is a zona pellucida. The zona pellucida is a glycoprotein polysaccharide-rich structure which must be penetrated by sperm and must be shed after fertilization occurs prior to implantation. A Graafian follicle is a definition of a secondary follicle that basically is about to be ovulated, and this follicle is large enough such that it extends to the entire cortex, and its structure is kind of illustrated in the micrograph on the next page. You can see basically in the center of the follicle the actual secondary oocyte. It's surrounded by its corona radiata cells. The area at the asterisk indicates the location of the zona pellucida. The granulosa cells you can see are outermost in this follicle, and they're separated by a basement membrane from the more external theca interna cells, both of which are obviously secretory in nature. And then finally, if we look over here, we can again see the stages across of how a primary follicle undergoes its development, the development of the secondary follicle, which will mature and form its Graafian stage. Recognize then what is actually ovulated. What is actually ovulated is just a secondary oocyte with its surrounding glycoprotein coat forming the zona pellucida, but recognize that that secondary oocyte is arrested in the metaphase stage of meiosis II, and only if fertilization occurs will that secondary oocyte complete meiosis II. So the answer to what is ovulated is a secondary oocyte arrested in metaphase. And the other question that's raised down at the bottom of this page is, where is the ovulated ovum immediately after ovulation? In other words, once it's extruded through the cortex of the ovary, it basically is now inside the peritoneal cavity, because there is a discontinuous duct system that does not actually directly link the ovary with the entrance of the fallopian tube. So on our next picture, we can see the proximity of the opening, the infundibulum, and its ostium, and its close proximity to the ovary, such that the ovum must pass briefly through the peritoneal cavity to reach the ostium, the opening of the uterine tube. So the infundibulum is the dilated distal portion of the tube. The portion of the tube that's longest and has the most clinical significance is the ampulla of the uterine tube. It's basically the most common site of fertilization, where the sperm penetrates the zona pellucida. It's also pathologically the most common site of an ectopic implantation into the wall of that uterine tube. Also shown is the point where the isthmus of the uterine tube enters the uterus, directly at the juncture between the body and the fundus of the uterus. And note that obviously the distal part of the uterus, its cervix, is inserted into the upper portion of the vagina. At the level of the cervical vaginal junction is what is known as the transverse cervical or cardinal ligament. This is largely connective tissue. It's traversed by the ureter, which lies perpendicular to the uterine arteries, but it's thought that this transverse cervical ligament helps prevent uterine prolapse, basically in postpartum females. The epithelium of the vagina is worthy of note. It's a stratified squamous epithelium that contains large components of prominent amounts of glycogen. Natural bacteria basically in the vagina break down that glycogen and convert it to lactic acid. It's that lactic acid that prevents proliferation of bacteria that have been introduced from the outside world into the lumen of the vagina. At ovulation, basically an increase in antral fluid pressure causes an increase in pressure on the wall of the follicle. Eventually the follicle ruptures and the ovum, along with its covering of the zona pellucida and its corona radiata, passes out of the ovary, and it must be fertilized within 24 hours or it's going to degenerate. The remaining portion of the Graafian follicle forms the corpus luteum, which we'll see is going to be converted to producing progesterone to help maintain basically the luminal epithelium of the uterus. So the follicular granulosa cells basically in that corpus luteum are the ones that actually begin to produce progesterone. If the ovum is not fertilized, basically the granulosa cells in the corpus luteum will stop producing and progesterone if they're not maintained by human chorionic gonadotropin, and it'll cause basically that corpus luteum to begin to degenerate about 7 days post-ovulation. And basically, if that ovum is fertilized, as we pointed out, the corpus luteum basically will increase in size for the next 3 months, and basically its progesterone output is then maintained by human chorionic gonadotropin that's produced by the syncytiotrophoblast cells in the developing embryo. And following about the 40th day of pregnancy, the placenta takes over progesterone function such that the corpus luteum, basically, its functional role is decreasing through the balance of pregnancy. That provides a brief summary of the elements of the female reproductive system, again concentrating on the functional significance of the stages of oogenesis that occur at the level of the ovary, what is actually ovulated, what kind of oocyte is actually present at birth, and basically the course taken by the ovum, fertilized or not, as it traverses the length of the uterine tube, uterus, and vagina, and pointing out the emphasis and the importance of the ampulla of the uterine tube as being the major common site for fertilization, as well as the most common site for the development of an ectopic implantation.